Good day, everyone. I'm Dr. John Ward. I'm the director of the Coalition for Global Hepatitis Elimination at the Task Force for Global Health. I'm also affiliated with the National Cancer Institute at NIH, and it's my pleasure to introduce you to and welcome you to the next edition of our NIH webinar series of moving from hepatitis discovery to elimination, where we look at different aspects of the NIH research portfolio that's helping us advance progress toward the elimination of hepatitis B and hepatitis C. Today we'll be focused on looking at different aspects of controlled human infection model, or CHIM, for HCV vaccine development. This will go from 10 a.m. Eastern time to 2:30, and as you'll see here in a moment, we have a very rich array of speakers and panelists to look at the different aspects of CHIM for HCV vaccine development. This is part of a long-standing webinar series that we do as the coalition with different institutes and offices of NIH. The webinar series, the presentations, the video recordings, synthesis reports are available on the coalition website at globalhep.org, and as this will be the case for this webinar series as well. I want to thank the organizing committee that puts together the webinar series each year. Liang, who you'll be meeting in a moment, is a member of that committee. And we put together a very rich array of webinars on different topics of research at NIH, and I really encourage you to go to globalhep.org and explore those if you're not already familiar with them. For this particular webinar, please submit questions to the Q&A function here on Zoom, and we'll do our best to answer them either as we move along for questions of clarity, and for those that are relevant to the discussions, we'll do our best to see how we can integrate those into the panel discussions. And as I've already mentioned, the webinars are recorded and are posted as shown here, along with the slide presentations. And if you want to stay abreast of the upcoming webinar editions, just sign up for the coalition newsletter when you go to globalhep.org. The coalition we started about 4 years ago this month. We function in 3 areas, bringing together the key stakeholders on relevant aspects of the services needed to achieve elimination of hepatitis B and hepatitis C. We are a source of credible information in a variety of different ways. And then thirdly, we have an advocacy or awareness raising function to really bring more capacity to bear on the different services that need to be offered around the world, including the United States, to achieve elimination goals. And I think this webinar fits perfectly with in each of those 3 functional areas. And indeed, together with the reliable tests we already have, the curative treatments, having an effective vaccine would truly be the final nail in the coffin for hepatitis C to be a ongoing health threat for human health. So it's really a fantastic area to explore, as we're about to start right now. So moving on, it's my pleasure to introduce Dr. Francis Collins, who's currently the Senior Investigator for the Center for Precision Health Research. But most notably, he's the former director of NIH, and indeed the longest-serving director for 12 years. He has a number of achievements in science in his own right. Has received the Presidential Medal of Freedom, as well as the International Medal of Science, and currently is the champion of developing a national hepatitis C elimination program in the United States. And it's indeed my pleasure to introduce Francis to open this discussion. Francis?
Thanks, John. It's a privilege to be able to speak to this distinguished group and all of the people who are listening to the webinar. I see 150 is the list right now on the participant button here, which is impressive. And of course, we're joined by a remarkable group of leaders in hepatitis C research, including a couple of Nobel laureates. So I know this is going to be a really interesting and productive day. I am still serving as the White House lead on an initiative to try to eliminate hepatitis C in the United States, taking advantage of the fact that we have highly effective oral agents with 95+% success rates with just one pill a day for something like 8 to 12 weeks. And yet that is not reaching the 2.5 million people who are currently already chronically infected with hepatitis C and who, as you know, face a potentially very serious outcome if the cure is not made available. There are a combination of reasons why we haven't been able to make that link. The administration, led by the president, strongly seeks this as an opportunity to do something historic and to figure out how we can get those direct-acting antivirals to the people who need them. So there is a proposal that the administration has put forward, which is currently being debated in the U.S. Congress. It has 3 components. One is to try to do a better job of developing the kind of testing capacity to enable test and treat in a single visit with a point-of-care finger stick test that would give an answer about viral RNA in an hour or less. Because we know from the experience that's been happening over the past few years that we lose a lot of people in what is currently a system that requires as many as 3 or 4 visits before starting therapy. And for marginalized communities where getting access to the clinic may not be so easy, that is something we ought to be able to fix. That is actually moving along quite well, taking advantage of the RADx program that was designed during COVID to accelerate point-of-care testing and applying it to hepatitis C. A second component is to try to reduce the overall cost per patient of what is still a course of therapy over 8 to 12 weeks that runs to $20,000 to $24,000 and makes this rather difficult for Medicaid programs and for programs that treat the uninsured or people in prisons to be able to do this. The strategy is to follow the lead that was piloted in Louisiana, the so-called Netflix model, to negotiate with the companies, Gilead and AbbVie, for a flat fee about which much conversation could be had about what would be fair. This has to be kind of a win-win. But once that has been negotiated, then the drugs would be made available for free to anybody on Medicaid, anybody uninsured, anybody in the prison system, anybody on the Native American reservation. And this would also capture lots of people in opioid treatment programs where we know the hepatitis C positivity can be quite high. The third component is to try to provide then better means of healthcare delivery to those marginalized populations, including some creative things such as greater use of telehealth and mobile units and other ways to get access to individuals who currently do not have access to good healthcare. And that's all folded into this proposal that was announced on March the 9th. It is now in the hands of the Senate in particular, where there's a lot of interest to turn this into legislative language that will capture those various components. We do expect that language to emerge potentially in the next 2 or 3 weeks. At that point, then it will be possible for the Congressional Budget Office to do their thing of assessing exactly what is likely to be the cost of this proposed 5-year program. We are optimistic that they will come out with a conclusion that indicates that this is not actually costly, it's cost savings because of the healthcare cost reduction that will happen by preventing many cases of liver cancer and the need for liver transplant. Our own economist, Jag Chatwal at Harvard, has done an analysis which is published in a preprint server, which looks awfully good, saying that even in the first 10 years, which is what CBO cares about, the federal government alone would save $13.3 billion in healthcare costs if this program was initiated now. And we don't believe it'll cost that much to run the program. So this should end up in a good place. CBO has been talking to Dr. Chatwal, so there's already a conversation going on about that. But in order to get over the finish line, this is going to take bipartisan support in both houses. Right now, we have more action going on in the Senate than the House. The House, of course, just came back from August recess and are a bit distracted right now. But there are some supporters in the House that have emerged, particularly those who know something about this disease. And have to mention Hank Johnson, who is a Hep C survivor, as one of those really powerful spokespersons. And on the Senate side, Senator Bill Cassidy, being a liver doctor who had a lot to do with that Louisiana pilot, is also a strong source of support and has certainly been talking this up with his colleagues. But it's going to take a big push. The advocates have organized. There are 126 organizations that have joined together, sending letters to the Hill and making visits to members of Congress to raise consciousness about this. But I think it's still the case that most members of Congress and most of the public haven't quite heard about this historic opportunity to actually do something we don't get to do very often, which is to take a terrible disease and reduce its incidence so much to the point that new cases will also start to drop in their frequency. And there's a lot of analysis of what that would look like. I'm fortunate in my efforts here to be assisted by Dr. Rachel Florence, who many of you know, who's my senior advisor and continues to put 100% of her time on this, but also now to have the assistance of Doctors Josh Sharfstein and Risha Irvin of Hopkins, who are now on IPAs to NIH to assist with this part-time and bring a wealth of experience about how to get things like this done. All that being said, if we're successful and if this finds its way into the budget for next year, and that is our goal, to try to have something ready to fold into that very messy process called the omnibus in December, which we still hope will happen. And then this could, in fact, get underway fairly quickly, because there's a very detailed plan and a strong support across HHS of all of the agencies that would be necessary for implementation. Everybody's excited about the chance to do this. But none of that will eliminate the need for a vaccine, which is what you all are here to talk about. In fact, we'll see what the legislative language looks like. The president's proposal includes some money to actually speed up and accelerate the search for an effective vaccine, because we know that could use a boost. We'll see whether that makes it through the congressional filtering, but that would certainly be an important part of this. Because as we all know, getting cured of hepatitis C with a direct-acting antiviral does not prevent you from potentially being reinfected. And if we really want a solution, not just for the US, but the whole world, that's what we need. And that's what you all are there to talk about today. And I wish you Godspeed in figuring out how best to achieve that goal with new technologies like mRNA, perhaps providing a boost in the opportunity, and the discussion you're going to have today about creative ways to do testing, perhaps involving volunteers. So thanks for the chance to say a few things before you kick off the real part of the meeting, and I wish you all the best.
And we wish you all the best in that effort, Francis. Thanks so much. Let me turn it over to my longtime colleague, Jake Liang. Jake is currently the Chief of Liver Diseases Branch and Deputy Director of Translational Research of NIDDK, and has been at the forefront of this development of a CHIM model for HCV vaccine development. Jake?
Thank you, John, and thank you, Francis, for your introductions. Let me go ahead and share my screen. Okay. All right, let me put on share mode. Okay, great. Well, hello, everybody. Welcome to this web symposiums. Thanks to Francis for sort of laying the ground for the need for developing a vaccine. So I don't need to speak more on the importance of HCV vaccine in achieving the goal of HCV eliminations. For various reasons that I don't have time to go into, efforts to develop an effective HCV vaccine has stalled over the last decade. We are not here today to discuss fully the pitfalls, barriers, and strategies in HCV vaccine development. Suffice to say, without a convenient animal model to test the efficacy of a vaccine candidate, we are doomed to falter. In this symposium, we are raising the prospect of a controlled human infection CHIM as an alternative to fast-track and promote HCV vaccine development. CHIM model is a model in which human volunteers are deliberately infected with infectious agents for the purpose of testing the efficacy of a candidate vaccine for this agent. This concept is not new. It was first applied by Edward Jenner more than 200 years ago in developing the smallpox vaccine. It has subsequently been applied to other infectious agents, including yellow fever, cholera, malaria, influenza, and dengue, and now to more than 25 infectious agents. I also want to mention that for the cholera vaccines, all efficacy data that serve as a basis for regulatory approval were obtained from CHIM studies. When the CHIM concept for HCV vaccine development was first presented to the HCV community a few years ago, people look at me incredulously with the exclamations of, "You're trying to do what?" Probably because we were so used to the idea that HCV is a terrible disease, and why would you want to do that? Many of us have spent our entire career trying to understand and beat this virus. I can understand why such a concept is contrarian to what we have all been working toward. Perhaps the rapid advances, as Francis mentioned, in highly effective HCV treatment have changed all that perception to date. The point is that we need an HCV vaccine, and can we leverage this sea change events to achieve these goals? And it was in late 2020 and beginning of 2021, if you all remember, that was a height of the COVID pandemic, and we were all shelter in place and couldn't go anywhere, couldn't do our work. So if this concept was really sort of came up, and a few of us figured out, maybe since we couldn't really do anything else, why don't we organize a workshop to really discuss, at least broach the subjects with the scientific community. And so this is really the genesis of the first workshop, and I wrote to all my colleagues in the HCV community, and they all thought, hey, you know, they were chomping at the bits. Hey, I really, same thing, we're not really doing anything else, so why don't we get together and just start discussing the feasibility of this model. And this led to a sounding board opinion piece by myself, Jordan Feld, Andrea Cox, and Charlie Rice in New England Journal of Medicine. And then we thought we probably should follow up with a more detailed workshop, and that was the genesis of the second workshop held in Toronto in 2022. So the rationale for controlled human infection models, not only because we really don't have a convenient animal model to test the efficacy, but also there are several other reasons. First is to accelerate early vaccine development. This model can de-risk vaccine candidate selections with early efficacy and safety testing before large late-phase trials. It can also reduce costs. The second is that this model can inform vaccine development and designs because of the detailed immunological, virologic studies and clinical correlate that can come from some of the early CHIM studies. And finally, this model can be used to assess promising vaccine candidates as a possible pathway to regulatory approval. As I mentioned, the cholera vaccine was developed entirely from CHIM's data. And also, when you look at the sort of the large clinical trial, such as the VIP trial to test the adenovirus vaccines, if we use this model correctly, we can actually generate more infection cases to determine efficacy than a traditional field trial for vaccine efficacy, such as the VIP trial. Because if you look at the VIP trial, when after they recruit 600 or 700 patients, only probably about 75 incidental HCV infection cases, and only 45 of them were used for final analysis. So we can easily achieve that number in a well-controlled monitor CHIM study. So obviously, we need a systematic approach to look at this particular question of controlled human infection model in HCV. Many of you, you're going to hear from many of the speakers and panelists about all the different aspects of this systematic approach. I'm not going to steal their thunder, but just want to quickly highlight some of the important points. Obviously, the clinical parameter, such as a high DAA response, really sort of the initial stage that we potentially could proceed with this model. Although the rate is high, we need to think carefully about how some of the patient may not respond, whether they will respond to a second treatment, and that will also be addressed in another talk. Another concern is that even with such acute infection, we can cure these patients. What are the long-term risks of acute HCV infection? That will also be discussed in the talk. And clearly, ethical consideration is going to be critical. we have engaged in several ethicists to discuss this issue. The importance of social value and potential risks are very important in determining whether this is ethically appropriate to proceed. And certainly we need to pass regulatory agency. We have begun discussion with US FDA and Health Canada about what the concern of the regulatory agencies are. And clearly the source of inoculum would be important to how we're going to use that in our CHIM model. And finally, clinical trial design is probably very critical in how we can design ethically, appropriately, fully informed clinical study to apply this CHIM model for vaccine development. Obviously, we need to do it in various stages, and starting with inoculants and maybe some sentinel cohort as well, and then eventually to vaccine testing. Again, this will be the focus of another talk later on today. So after the Toronto workshop, and a few of us thought that, well, why don't we put all these concepts and discussions and all the nuances on CHIM model for HCV on paper. So we have recruited various different investigators. Many of them will speak in this symposium as well as panelists. And so we put together the supplement that's just published in Clinical Infectious Disease that really cover full gamut of the different topics. So although this symposium will be used to discuss a lot of these important points, so if you're interested in looking at some of the details, I will refer you to this a link here that will be also flashed in the chat box. So feel free, and all the articles are open access, so everybody can look at it. So now today really comes down to today. We thought maybe it's a good way to get all of us together to really talk about it and to have a real conversation on the potential and feasibility of the CHIM model. So we're going to begin with a talk on the ethics, and then moving on to taking a perspective from the participants, and then we'll follow with a panel discussion on these couple issues. And then the next session will be on the clinical parameters, such as the DAA treatment for acute and recent HCV infections, as well as addressing the long-term consequences of result acute HCV infections. That'll be followed by a second panel discussion, and then we'll take a necessary biological break. And then moving on to really, I consider it's a really important part of the symposium to look at what particular would be feasible in terms of the challenge inoculum for HCV CHIMs, followed by a clinical trial designs, and end up with a various different basic science study that we can do, since the model really offers unprecedented opportunity to study HCV infection. And then we'll end with the last panel discussions. And we'll also do something fun. We'll ask 3 questions to the audience, so everybody can chime in and take a poll. And I think and these are sort of important questions. We won't reveal them yet. We will reveal them at the beginning of the third panel. So if you do stay around, please chime in and offer your opinions. It'll be totally confidential, very simple questions, and yes and no, maybe answer. So you can click on it, and then we'll share the results with you at the end. So without further ado, I will stop sharing my talk, and moving to the next speaker will be by Dr. Charles Weijer. He's a professor of medicine and philosophy at Western University, London, Canada. Unfortunately, Charles won't be able to present in person because he has COVID, which shows that really COVID is still around. And he was actually quite thoughtful when he was first diagnosed with COVID a few days ago, because he was exposed to somebody who had COVID. He thought, well, he probably should put a recorded talk together in case he couldn't make it. And so he prepared to send it to us. We kind of was hoping he would be able to join us as either as a panel discussion or present, but unfortunately, he is somewhat symptomatic, so he preferred not to join. So please join me in wishing Charles speedy recovery. And certainly we're very thankful for him to put together this recorded talk. And otherwise, I will have to step in to talk. You probably don't want to hear me talking about ethics. So without further ado, let's go ahead and share the recording.
Thank you very much for the invitation to speak with you today. It's a pleasure to speak to you about
The volume
ethics
is a little low.
of
Can somebody
hepatitis
turn it up?
human infection studies.
This is, I think, the top level for the video, so just please turn it up on your computer so you can be able to hear.
Okay, thank you.
More than 20 infectious agents have been researched in controlled human infection studies over the past decades. These infectious agents cause a spectrum of human disease, from the benign, such as the common cold, to the serious, such as malaria and cholera. Controlled human infection studies, or CHIMs, have led to important knowledge gains. They've played a role in the development of vaccines to prevent cholera, dengue fever, influenza, and malaria. And modern CHIMs have a remarkable safety record. And this is, I think, a testament to the lengths that scientists go to protect healthy volunteers in these studies. For example, of 6,000 participants in malaria and influenza studies, only 4 serious adverse events have been reported. And no one has died in a modern CHIM study, not one person. So, would it be ethical to intentionally infect healthy volunteers with hepatitis C in a CHIM? I think until the last few years, the idea was kind of unthinkable. Writing 2 decades ago, Frank Miller and Christine Grady wrote that CHIM studies were on a spectrum, arrayed from clearly acceptable on one end to clearly unacceptable on the other end. They wrote that clearly unacceptable would be controlled human infection models in which infections are induced for which treatment is non-existent or ineffective, symptoms are intolerable, or serious morbidity or mortality is likely to result. For example, hepatitis C. But circumstances have changed with the development of highly effective direct-acting antivirals for the treatment of hepatitis C. And I think it's now timely to ask, are hepatitis C CHIMs ethically permissible? In a paper published last month with colleagues, we set out a framework for the ethical conduct of such studies. Our 10-point framework sets out key ethical considerations for the design and conduct of controlled human infection studies with hepatitis C. We believe that studies that satisfy our 10-point framework are ethically permissible. In what follows, I'll briefly discuss each ethical requirement. First, hepatitis C CHIM studies must have sufficient social value, and this means that CHIMs must generate knowledge likely to be used to advance the goal of global elimination of hepatitis C. Despite the existence of highly effective direct-acting antivirals, treatment as prevention has faltered globally due to limited access to hepatitis C diagnostics and treatment, and unfortunately also due to high reinfection rates. Plainly, a vaccine is needed, but development is currently hindered by high costs, the lack of an available animal model, and the practical challenges of conducting a field trial. CHIM studies can provide preliminary efficacy data, allowing researchers to prioritize vaccine candidates for field trials. And efficacy data from CHIM studies themselves may inform regulatory approval of a novel vaccine. Second, studies must have a reasonable risk-benefit profile. This means that risks to participants and third parties must be minimized, and risks must stand in reasonable relation to the social value of the study. The risks of acute infection with hepatitis C are quite low, Most people are asymptomatic, and the risks of severe acute hepatitis is less than 1 in 1,000. Further, direct-acting antivirals are highly effective with cure rates approaching 100% in this setting. Participant safety can be augmented through study design. In the first stages of the development of a controlled human infection model, the first participants should be infected and treated sequentially to ensure that people can be infected safely and treated successfully. Further, the safety of a 3-month delay in treatment should be established before increasing the duration of untreated infection. Third, hepatitis C CHIM study sites must be carefully selected. CHIMs should only be conducted at hepatitis C referral centers with the clinical expertise to care for patients and treat any complications. Further, sites should have a track record of conducting high-quality clinical trials. And finally, sites should have a strong relationship with the community, including people living with or with experience of hepatitis C and people who inject drugs. Fourth, participant selection must be fair. Participant selection should prioritize minimizing risk to participants and third parties. To minimize risk to participants, they should be healthy volunteers, 18 to 40 years of age, who lack comorbidities or liver risk factors. Participants must, at the time of enrollment, provide written consent to direct-acting antiviral treatment upon study withdrawal or completion. Risks to third parties must also be minimized. Participants must agree to notify intimate partners and use condoms for sexual intercourse until virological cure has been documented. People who engage in behaviors associated with transmission of hepatitis C should be excluded from CHIM studies. Fifth, hepatitis C CHIM studies require robust consent procedures to ensure informed, voluntary, and comprehending consent is obtained from competent adults. Information should be communicated to prospective participants using evidence-based methods to promote understanding. Researchers should partner with clinical educators and stakeholders to develop educational materials to support the consent process. Prospective participants should have the opportunity to discuss the study one-on-one with research staff. And the prospective participants' comprehension should be formally tested. Any gaps identified should be addressed through education and retesting. Okay, so those are the first 5 requirements in our framework, and this is a brief visual interlude. So let's now carry on to the final requirements of the framework. Sixth, participants in studies should be compensated. Participants ought to be reimbursed for reasonable expenses, including childcare, parking, and transportation. Further, participants ought to be compensated for time spent performing study activities, such as attending clinic visits. Being a study participant is akin to unskilled labor. Accordingly, participants ought to be paid at the locally applicable hourly minimum wage for unskilled work. Consideration may be given to modest compensation in addition to this for the time that participants spend infected. Additional financial incentives, though, we think are best avoided. Seventh, hepatitis C CHIMs should engage relevant stakeholders, both to inform the study's design and promote transparency and accountability. Achieving the ultimate goal of hepatitis C vaccine development requires engagement with researchers and pharmaceutical companies developing vaccines, as well as regulators on scientific aspects of the CHIM. Engagement with prospective participants, including people who've participated in other CHIMs and advocacy organizations, may usefully inform study procedures, as well as recruitment and consent materials. Finally, as future trials of vaccine candidates will require community support, it's critical to engage early people living with or with experience of hepatitis C and people who inject drugs. Eighth, studies should be conducted in fair and open scientific partnerships. Researchers should publish the protocol for these studies as soon as possible. Once established, researchers should make available the inoculating agent to qualified scientists at other centers who are planning to conduct hepatitis C CHIMs. Finally, researchers are encouraged to agree on a common set of outcome measures and share their own data to facilitate pooling data across studies. Ninth, hepatitis C CHIM studies require independent ethical review and oversight. Studies must, of course, be reviewed and approved by a research ethics committee. The Research Ethics Committee should have experts in the clinical care of hepatitis C, as well as representation from relevant stakeholder communities. Hepatitis C CHIM should be overseen by an independent data and safety monitoring board, and researchers should consult the Data and Safety Monitoring Board at major decision points, including escalation of viral dose and extending the duration of the untreated period. 10th, and finally, research ethics. Ethics research should be integrated into ongoing studies. Integrated ethics research can be used to make improvements in the study. For instance, knowledge gaps in the evaluation of comprehension of consent information may suggest changes to educational materials, or interviews with participants may suggest changes to the frequency of study visits and the adequacy of compensation. So we conclude that it can be ethically acceptable to develop a hepatitis C controlled human infection model. Indeed, when done appropriately, developing such a model should be a priority on the path towards global elimination of hepatitis C. I'd like to thank my co-authors and colleagues Annette Ridd, Jordan Feld, and Jake Liang. Thanks very much.
Okay, well, thank you very much, Charles, for presenting, despite the fact you're not here. We appreciate that. And let's move on to the next presentation, and who is Jake Ebers. He's a communication director of the One Day Sooner organization. He's a community activist as well as advocate. So we very much look forward to his perspective from a participant point of view. Jake, please proceed.
Thanks so much. I'm going to go ahead and share my screen. Fingers crossed that it works out seamlessly. Awesome. Are we good? All right. So hi, everyone. My name is Jake. I'm super excited to be here today and kind of talk a little bit about the paper that I was the author on, along with other participants, former participants in human challenge studies. I want to start out here and again, thanking Jake Liang and everyone else for organizing this effort. And want to highlight, I am one person. We have kind of survey data as part of this paper, but my position is, and One Day Sooner's position is, just one of many. But we do think that it is kind of worth sharing when it comes to volunteering for infection. So a little bit of background about us really quick. We are a nonprofit that advocates for healthy medical research volunteers, particularly people in challenge studies. We've been independently collaborating on these proposed HCV CHIMs. And among our membership base include over 40,000 people across the world who have at some point expressed a specific interest in participating in a CHIM, whether that's for any range of diseases. And about me, I'm the communications director I'm also a former Shigella challenge participant. Shigellosis causes dysentery. My mother is super proud because this is kind of the outcome. Kind of sensationalist headline, but very positive article. And I'm very, very proud of my participation. This was at the University of Maryland. I contracted dysentery to help test a vaccine. And it was not fun. Dysentery is not fun, but I am very proud of, personally proud of my kind of contribution to society. And hopefully one day, having gotten sick with dysentery means that others won't in the future. So the paper that we wrote was called Volunteering for Infection, Participant Perspectives on a Hepatitis C Controlled Human Infection Model. 4 of the 5 of us are former human challenge study participants for malaria, COVID-19, norovirus, and in my case, Shigella. And then it was a survey of our membership base from English-speaking countries where CHIMs are commonly run. And the number of people who passed this, the comprehension questions was about 117. Most of these people have not done CHIMs before, but have expressed pretty strong interest in them. So this is not a representative sample of the entire population, but it is an interesting group to kind of think about the problems and issues and potential of HCV CHIM with. We introduced the concept of the HCV CHIM and asked them a whole range of questions on their concerns and thoughts and pretty much any feedback they had. Overall, there was pretty strong support for the concept. This is not particularly surprising, given that this was a group that is predisposed to being kind of favorably oriented towards them. And people were generally in the abstract kind of interested in the idea of volunteering. And so what this kind of like phrases is there's this, group of altruistically minded people who would want to participate in this sort of thing. And the question then becomes, how do you make, best use of this group? And when you're designing and implementing a challenge study model, right? So I'm going to start with some of the concerns that were raised. And one of the first ones is not surprising at all. The greatest concern was over long-term health effects, right? And there was a strong desire for people to know about their health measures during the study, and that would require, things like your bilirubin count, but like kind of, explaining that in the context of normalcy and what that means for your body in the long run. There was lower enthusiasm for longer periods of infection. It's not sure, we're not clear if that's because of just inherent hesitancy or just what that means in terms of longer timelines. But so there would need to be, and the kind of the median sort of months we presented on a scale or chosen by the survey participants was about 3 months. There are plenty of people who went beyond that and people who had lower thresholds, but that was just something to note. 53% of respondents were pretty enthusiastic and said they would even consider participation if they were found to be at slightly higher risk of complication. Obviously, Charles pointed out that you would want to select for people who do not have, pre-existing conditions. And so I don't think it'd be a good idea to utilize this kind of group. But what this does point out is that there are people who do have a higher risk tolerance threshold if they believe that it is potentially worth it for the greater good. The altruism that's present here, and that is, is, I think, pretty important, right? So our survey showed that altruism is a pretty strong factor driving people to participate. And that is something that's reflected in medical literature about healthy human volunteers more generally. There's a spectrum of motivations, but altruism tends to be one of the primary ones. And yeah, the altruistic motivations among our survey were to advance medicine and to save lives. And that was, those were the 2 highest ranked motives. And so I would argue from this that scientific utility and impact is thus morally relevant. And what I mean by that is, if I am volunteering my body and my health temporarily to, in hopes of one day making, for instance, a vaccine or contributing to the process that would create a vaccine, then you are basically taking advantage, in a good sense, of my altruism. And I would argue that the subsequent moral onus is on you to make sure that you are doing so in a scientifically valid way, right? And that means that there are some situations in which, particularly if a participant or group of participants, or if participants are concerned with, scientific utility and helping people, there might be a world in which some sort of study design decisions that have, relatively minimal increases in risk, for instance, might not be inherently objectionable if they yield sufficient scientific value. And that means that things like model quality are also pretty important, right? If the CHIM takes 10 years and doesn't actually prove pretty competitive with a field study, I would argue on another level too, that kind of runs counter to the interests of volunteers themselves. And so things like the speed, the ability for the model to be adopted, the capacity of the model, and the kind of site for regulatory pathway, those are all morally relevant factors, I think, when we consider the altruism of healthy human volunteers. And then there's also the transparency aspect. So I think there's an ethical obligation to ensure that the data generated from these bodies of volunteers is made useful to the public to the extent possible, right? One Day Senior is currently working on a transparency pledge for HCV CHIM, which many of you will be hearing more about later. But I would argue that, again, this ability to contribute to the greater good is a benefit of participation for many people, or at least a soft benefit. It is a reason that drives them to do so. And so one thing that I think is important is being upfront about why a study is important. And I think this is something that a lot of medical research kind of fails to do. And there are a lot of complicated reasons for this, but often it might just come down to what you often see as this idea that, well, this might help develop a vaccine someday, and kind of stopping there. There seems to be a hesitant to kind of describe wider benefits to society. And I would argue that, again, kind of runs counter to the views of volunteers that have been expressed both in this survey and in wider medical literature that show that people really do kind of take this seriously, many of them at least, and explaining that is in their interest. Sharing results is a very simple one. The simple write of email a year after the fact can be very helpful. And like I said, you can be a little more specific about the social benefits than, quote unquote, helping vaccine research. One thing that we touched on was compensation. I want to disclose here that this gets a little bit more into our authors' viewpoints than necessarily what was in this survey. We can say that the survey respondents were more concerned about underpayment versus overpayment when it came to compensation. Factors that were seen as relevant to compensation included time and inconvenience, which would be pretty high in an HCV challenge, but also things like long-term health risks, which would probably be pretty low, as we're going to hear from later speakers. Discomfort/illness, moderate. I say moderate because my threshold is dysentery, so I might have a weird sort of sense of what is acceptable, or at least personally tolerable. And then potential benefit to society, which is very, very high. So we kind of proposed a pretty high target for a 6-month induction period when it comes to compensation. At the end of the day, almost any sum of money, whether it's $2,000 or $20,000, is still a lot of money to somebody. So if we limit ourselves to a price ceiling of what is quote unquote reasonable, we do not solve what the problem of undue inducement, this idea that we might induce people to participate in a study for money only. We are just pushing that problem down the socioeconomic ladder. The key issue, I think, is ensuring that informed consent processes are sufficient, and that is independent of money. If your informed consent process is sufficiently robust, I would argue that you could hypothetically offer any sum of money, and that would not necessarily present an ethical issue per se. There are definitely reasons to avoid, for instance, $20,000 budgets are one of them. Too much money can actually signal to prospective participants that you think the study is risky when it might not be, or risky in a way that it might not be. But part of the argument we make in the paper is that we should avoid lowering compensation for the sake of lowering compensation. And then the kind of critique we had of the market rate sort of model was this underlying argument that unskilled labor is not necessarily paid justly or fairly in a market system. And so we want to be cautious about the degree to which we rely on that as a benchmark. So overall, what this survey showed was that a properly explained HCV CHIM is not outlandishly scary. There are a lot of people who would consider participation on altruistic grounds. And I think a lot of important background information that we provided in our survey about, for instance, the extreme difficulty of testing vaccines in the field among people who inject drugs, the really serious global burden, kind of nudges volunteers to decide why they might want to participate, because otherwise they don't quite have the background in the disease to understand the significance of, why a study like this might be really, truly important and transformative for the field. I would again want to emphasize that ensuring studies are efficient and scientifically useful is an issue of respect for volunteers as well. And that valuable contribution should be recognized. In our case, we think that does translate into higher compensation. And I want to emphasize too that I don't think altruism and compensation are mutually exclusive, right? I don't think we would say that firefighters, for instance, a firefighters union asking for a higher pay rate would be necessarily selfish or would impinge upon the kind of altruism that we associate with that profession. One thing we do bring up really briefly in the study, in the paper, is this idea of kind of splitting the difference, where, for instance, you double compensation, but you take that doubled half and have it have basically be a fund that can be directed by the volunteer to some sort of charitable institution. So there are a lot of ways to go about increasing compensation, but overall, there are ways that can also be a little bit more perhaps tangible or palatable to IRBs. So that is all I have to offer. I think I'm really excited, as One Day Seniors Communications Director to continue assisting with the development of this model. We think it's going to be very, very important. And we're super excited, as is our volunteer base, about the prospect of accelerating vaccine development for hepatitis C. So thank you very much.
Okay, thank you, Jake, for that wonderful presentation from the perspective that we often don't hear about and probably should pay a lot more attention to it. So again, let's go ahead with our first panel discussion, which would be led by Dr. David Thomas, who's a professor of medicine and epidemiology in Johns Hopkins Bloomberg School of Public Health and Johns Hopkins University School of Medicine. David, please proceed with the panel.
Yeah, thanks a lot, Jake, and to both of our presenters, thanks for those terrific talks. Jake, I'd love it if you stay on the panel, and we also are joined by Dr. Andrea Cox, who's a professor of medicine at Johns Hopkins. Jake Liang is going to participate. Kim Page, a professor at University of New Mexico, and Daniel Raymond from the National Viral Hepatitis Roundtable. Thanks to all of you for unmuting, for making yourselves visible so we can tell who's on the panel. And we'll kick off with, unfortunately, Charles won't be able to be on the panel for the reasons that Jake already mentioned, but let's go ahead and try to get into some of these subjects that both speakers brought up. Let's start out with this issue of compensation right off the bat. And Jake, you just said, that whereas the pure ethical consideration wants to keep compensation low to avoid unfair inducements to coercion, if you will, then the participants have a slightly different take on it, that actually that sort of thing can be managed in various ways. And I'm wondering, we have Donald Brown, who I didn't introduce, but who's had extensive experience running research studies and participating, and Daniel. I wonder if the two of you would want to comment on that issue, and then Jake, any other comments that you would want to have on this issue of how do you balance the right amount of compensation or reimbursement, if you will.
Well, for me, Dr. Thomas, first and foremost, good morning, and it is a pleasure to be a part of this panel discussion. How do you balance it? I always look at the amount of time, the amount of blood that would be required, and honestly, really appreciating the time that any participant put forth that we recruit brings to the table, and understanding that we have to compensate them for their time. It's been my experience over the last probably 20 years or more with conducting outreach in community, working with syringe exchange. Compensation for research participants have always been an issue. And that's the one thing that will continue to affect any recruitment trial moving forward.
Daniel?
I don't have a bright line here. I think there is a meaningful difference between compensation and inducement. That the level of compensation should not be perceived as an inducement that could be interpreted as potentially exploitative. This has obviously been a big debate in things like organ donations and so forth. So I think that there's probably an extensive bioethical literature out there, but compensation is certainly appropriate, but I think there's also perhaps a higher degree of challenge to ensure that the CHIM studies are seen as having integrity, because I think that there will be a considerable degree of scrutiny from people who are not necessarily familiar with this model and maybe bringing a various lenses to how to understand this as an ethical approach.
Yeah, thanks. I don't know if the other panelists want to weigh in on this. I'll just speak for myself, and I've heard and read ethical takes on this, and of course we're all familiar with Dr. Alter's work showing the huge improvements in the safety of the blood supply, switching to an all volunteer compared to a compensated donor pool. So, but I will say, when I read Jake and his coworkers' article and heard it from participants' side, and then this idea that getting Shigella or hepatitis would be compensated at a level of a minimal unskilled worker, it did feel a little like I really felt like that was a very important perspective to listen to, and to for us all to sort of adjust to hearing from the persons who might be so altruistic as to donate their bodies for this cause.
Yeah, if I may, I have a lot of I recognize that this is a really ethically fraught problem, and I don't have any sort of animus towards people who I think have done a lot of thought and have concluded that an implicit price ceiling, for instance, might be important. The kind of things I wanted to highlight were, I think, the contradiction of what I'm going to call the kind of price ceiling, right? Because I also think Dan is right. There's a public perception aspect at play too, which is a separate kind of issue, but related. But in terms of, for instance, so I take it kind of at a personal level. The Shigella study that I was in involved injection with an experimental vaccine and a 10-day inpatient, in addition to, drinking a little shot glass of the Shigella bacteria. That study originally paid around $3,200. And shockingly, very few people signed up, but some did. And they had to over double that compensation. I did not see that study until after that compensation had been doubled. But I can pretty confidently say that the original $3,200 was just not something that would have worked out for me. People exist on a spectrum. I have a colleague who is in one of the co-authors, Alistair Fraser Urquhart, was in a challenge study, was paid about £4,500 and donated every single penny of that to charity. I'm not nearly as good of a person as he is. But I think that the argument that became most persuasive to me was that, for instance, $3,000 or $7,000 is a lot of money to somebody, right? That is like months of rent potentially. So I was working a, office job in DC. So $7,000 was a lot of money, but not life-changing sums of money, but that is life-changing sum of money to somebody somewhere. So a price ceiling does not necessarily solve that problem. It just pushes it down the socioeconomic ladder. And then the final thing I try to bring up is, and I'll use the example of DC, the living wage in DC in May of 2022 was about $22.50 an hour. That's for a single person without children. The median, or like the 75th percentile, according to the Bureau of Labor Statistics, for a fast food worker, an unskilled laborer, a kind of the typified unskilled laborer, was about $17 an hour in the DC area. And so I think we need to be really careful about tethering pay to the market, because there is a very big difference that from a market rate versus what is a just rate. And I would argue that not paying a living wage in this case would, even though you're not trying to get people to live off of it, I think that's an example of, it kind of shows how relying on that market rate as a threshold is maybe a little bit more ethically fraught than I think it may be thought to be.
Thanks, Jake. And while you're still unmuted, I'd like to come back to and flesh out this other really important issue that you touched on, and that Charles did as well, which is ensuring that there is value to the study, that people are donating their bodies for with the idea that there will be some important scientific contribution. As being on the other side of the fence, we're just, I guess I'm starting to wonder, how certain would we need to be? Obviously, we'd want to ensure that a study was going to be have a high likelihood of giving a meaningful result. But others have brought up even more extreme views, that perhaps you need to have all the money in place for the phase 3. You need to have a company ready to say that they'll pay for ultimate production and worldwide distribution. You need to ensure access to low and middle income countries. Where do you think that, as someone that's donated your own time and body, where does the line finish? I mean, how much confidence would you need to have?
Yeah, I think that actually comes in, there are kind of two levels, I think, here. So there's confidence that within the study that you are designing, right, that it is being done to maximize potential scientific utility. I think that is pretty important. And I think, as I mentioned briefly there's this idea of people sometimes being willing to accept increased risk to reach that point, if it's properly explained. I don't think that should be discounted. A good example, if, for instance, when you're collecting the inoculum or the inocula for HCV CHIM, you could theoretically stagger it one person at a time for years, right? And that would technically provide a better safety profile before moving on to larger groups, but that would sort of defeat the purpose of moving quicker, right? And so at some point we do make implicit sort of or explicit calculations of what minimal increase in risk is possible for volunteers. At the meta level, I don't think you need to be like, I think it would be, that sort of wild goose chase, or I think it'd be impossible, right, to argue for, like, you could never do that for a vaccine study, right? You could never look someone in the eyes and say, 90% of phase 2 vaccine candidates fail. So in all likelihood, this one, like, you can't, I think within the study, sort of like the intra study kind of scientific validity and utility is really important. I don't think you need to lie or pretend that science is a predictable endeavor and that you can predict the future. There's so much that goes into whether a vaccine is effective, uptake, the other timelines. You can't predict that, and I don't think you need to. I think making a reasonable statement that you make to the IRB and regulators when you submit this study is fine. Saying, this could very much help move the needle forward on creating a vaccine. There might be a meteor strike in 10 years that destroys all biopharmaceutical manufacturing capacity. Having to kind of like line up every single duck in a row, I think is not, that's not something I think that volunteers would demand, and I don't think it's probably possible.
Yeah, I see Jake's hand up, and I will call on him and also encourage others, if you have something to say, to pop your hand up so I can recognize you.
Yeah, I like to explore this question a little further about the duration of the study, right? I mean, Jake, you mentioned that in your survey that most people thought 3 months was okay, but the question is really, if it's necessary to extend to 6 months for scientific justifiable reason to test the efficacy of the vaccine, do you think that make that big a difference to the volunteer, 3 months versus 6 months? Obviously, symptoms and other complications of being infected is a consideration, right? If you have to deal with 3 months symptom versus 6 months of symptom, I imagine when you did the volunteer study of dysentery, the shorter the better, right? So the question, but we all know for hepatitis C, most of the time it's very mildly symptomatic. Most of the people don't even have it. So is that a sort of a consideration for volunteers, if whether 3 months versus 6 months, that big a difference? And understanding that the longer you infect, the slightly increased risk you may have. But I think for a clinical study, 3 versus 6 months really doesn't have any difference in terms of clinical outcome. So I just want to kind of ask you from the sort of participant point of view, if we explain clearly to the participants whether 3 versus 6 months makes a bigger difference.
Yeah, and I think I think that is a very easy and compelling argument to make. The limitation of a survey like this is people are kind of going in. Once they reach that question, they have a kind of vision of the HCV CHIM in their head, and the kind of the inherent sort of framing issue of giving someone a slider with months on it. They tend to kind of go towards median, and we put 6 months as the maximum because that was what we understood as the maximum timeline. There were kind of sub questions that implied that people would be willing to, for instance, were more receptive to a longer timeline if there were some scientific value to it. So I don't think, I would say I don't think 3 or 6 months makes or breaks it. I think the compensation thing is helpful. Living with a stigmatized disease that requires significant changes to your lifestyle and significant burden, that is like a relevant factor, and naturally someone would want to do that for less time than more time, all else being equal. And so I think compensation can help offset that burden. But I think so much of it comes down to when we're talking about well scientifically explained concepts. I've read a lot of informed consent documents right now. I'm actually screening for another medical study, fun fact, which, disclaimer, is not like a requirement of my job. That would be ridiculous. It's personal joy. But so much of it kind of is presented as fait accompli, and there's not a whole lot of explanation of benefit to society or how the decisions that were made about, what my body would go under in terms of risk came about, right? IRB decisions are often, kind of a black box. You guys clearly put a lot of thought into this about what risk tolerances are acceptable, and shoving that all into an informed consent document might not be feasible, but showing the work, I think, is potentially very useful for people who are interested and might be interested. I don't know if that entails like an appendix saying like, here's the research, here's a summary of the research that made us think that 6 months versus 3 months is not a particularly significant difference in terms of your health risks, things like that. So yeah, bottom line, I think I wouldn't, I would not look at that 3-month median figure and be too afraid that no one's ever going to do 6 months. I think provided there is legitimate scientific purpose, which there is, then that's not going to be a huge hindrance. But it definitely, all else equal, will be harder because you're asking someone to do something for longer.
Yeah, thank you. And I do want to delve a bit more into the consent process and even the issues of whether, as was mentioned by Charles, that we need to have a test of comprehension and how that would work, that dynamic of how much to disclose, how much is comprehensible. Those are all, I'd like to hear other voices on that. But I see, are those the points that Andrea, you and Michael want to raise, or should we take your comments first before we shift more focus on the consent?
I'll defer to Michael and then I'll make my comments.
Michael.
Well, I was addressing one of the issues you raised, David, about are there companies out there that will take on development and delivery of the vaccine if a successful CHIM is demonstrated. So let me just translate it to a question to Jake. Great talk, Jake. Thanks for all of your tremendous efforts. But for the other CHIM trials against all these multiple pathogens, were there companies committed to developing the vaccine if and when the CHIMs are shown to be successful? Because with Hep C, it has been difficult to get the big vaccine companies to commit to developing a vaccine. I've had several interactions: Moderna, BioNTech, GSK, Sanofi Pasteur, and none of them, certainly up until today, to my knowledge, were willing to develop a Hep C vaccine. They did not consider it profitable enough.
What I can say really quick is that, the studies, most of these studies are not characterization studies, right? So the one I was in, for instance, was a vaccine candidate that had been developed by Institut Pasteur in France and the University of Maryland. And for malaria, for instance, a lot of the malaria CHIMs are sponsored by the various biotech companies that are trying to fight malaria. So I think, I can't speak to unfortunately, the wider kind of landscape, but I do, because usually once you get to the HCV, or once you get to the CHIM vaccination level, the answer, or the vaccine trial, the answer is yes. I think there's maybe an argument to be made that once the CHIM is established, that is a signal to the field that it's going to become a lot easier to test. And so that will kind of rev up the circumstances. Some internal research One Day Sooner has done has suggested that there are, and I'm sure you're probably even more aware of this than we are, there are well over a dozen potential candidates in the early stages. But yeah, so it's, I can't speak to the, in general, how CHIMs work like that in the early stage of a CHIM establishment.
Yeah. Well, it might mean that, public funding needs to play an important role to take the vaccine over the line. But to do that, get it through phase 3, normally, always that's done by private companies, to my knowledge. So I don't know, there might be an additional requirement from the governments of the world to combine funding to do this, because right now companies don't seem very motivated.
That's a great point, and I think that is why I brought that up. So we have Andrea, Harvey Alter, and then Susanna Nagy in that order.
Yeah, I wanted to address the issue that, we might need a company ready to pick up a vaccine at the end. And I do think that CHIM will probably follow phase 1s where people are not being challenged. So we'll have some sense that the immunogenicity of these vaccines is good. But one of the things that I think dampens enthusiasm for development of a vaccine, including from big pharma, is the notion that it's going to be exceptionally challenging to do. But I will say that taking the vaccine candidate that we did test, which didn't have a neutralizing envelope, it didn't have envelope and couldn't induce neutralizing antibodies, we saw what has not been seen in a single HIV vaccine trial to date, which is suppression of peak viremia in those who were vaccinated. So our first try, while the vaccine didn't protect, it did have an effect on the virus. And I think, as I said, that's not been the case in other vaccine trials. So I do think there's a foundation upon which we can build and create an effective vaccine. And I think that the more we develop things that induce immunogenicity that's robust, the more likely we are to be able to find someone who's willing to carry that forward. And I think that having a challenge virus that is homogeneous across recipients will help tremendously, because some people cleared the infection in the vaccine trial we did. Some people suppressed viremia, others did not. And the heterogeneity of the patient population plus the viruses with which they were infected adds complexity to that analysis that could be eliminated by the CHIM. So just as Jake Liang said in his remarks, the ability to control some of those variables will help us to understand how to make better vaccines, and as well as making them quickly with the CHIM.
Thanks, Harvey, then Susanna, and then Donald, and I think we'll be out of time.
Yeah, thank you. Michael's raised an interesting point, but I think we need to divorce the concept of having a company ready to go from the concept of CHIM. CHIM is a preemptive step, a unique kind of thing that we're actually getting ready for what we hope will come. And since we know that the vaccine is what we really need, that the test and treat strategy will work in some areas, but it's not going to be a universal cure. And we do need the vaccine, and so we need to get ready for it. When somebody is ready to have the vaccine, we have to have a way to test it, and that's what this is all about. So everything Andrew just said is valid and true, and I think we're on the right path. This is really kind of unique to plan ahead, be ready to go when something good is there. And on the other hand, I think having the CHIM model and the right inoculum will help to induce companies to go this very costly path and shorten the clinical trials and all that. So I think we're on the right track.
Thank you, Harvey. Susanna.
Yeah, so Dave, I was going to go back to the informed consent issue, if that's okay for a second. And there are probably folks on who are experts in this area, but I think this is a great opportunity to think about engaging the community and how they would want to consume that type of information. I think it provides an opportunity to think about an informed consent process that does not involve a 25-page written document. And I think it's going to be really important to understand what pieces of information and in what way should that be delivered to potential participants so that they can make the most equitable and fair informed decision.
Yeah, that's actually exactly where I was going to go with that, and I appreciate you bringing that up. Donald, you've done a lot of informed consent over the years, and you've used a variety of instruments. Did you do it for the VIP trial?
Yes, we did. And
can you comment on how that went? Do you feel like how do you feel about that?
Well, I was just going to piggyback on what Suzanne was stating, is that take it to the community, do some focus groups, engage the community. And one of my concerns was with the target population. When you talk about 18 to 40, I know that would be challenging, as you're not seeing many young adults transition into injecting drug use. But I know focus groups has been paramount when we bring anything to community, we need to get the feedback from the community before we launch any community trials. I know I honestly can say I have an excellent, me and the team, we have an excellent relationship with community engagement of stakeholders in the community, and we have to take it to the stakeholders, to those who we wish to include and bring them to the table so we can get their feedback on what is appropriate, as well as compensation for such a relative trial. I think it's an excellent trial. It's something that's long overdue, and we know how desperately we need it, but we cannot move forward without community input from all those involved.
Mm-hmm. So Donald, would that mean, just to push that point, let's say that all of us, the doctors people, would say, oh, there might be 1 in 10,000 risks that somebody might have a higher chance of getting a cryoglobulin vasculitis. Okay, some big word like that. Like, would that be something worth trying to explain in 2 pages on the consent form? First, what is it? Then what is 1 in 10,000? And then testing to see if there's comprehension? Or do we leave that kind of detail, as much as the lawyers would like to have us all have it in there? Do we leave it out?
We leave it out. We leave it out. We're going verbatim for the consent, and then we're going over that consent with the client verbatim. And the longer consents, we have a tendency to lose those clients. You know, we have a tendency to lose them. So, you know, as much as we know the importance on the lawyer side is critical, I would say leave it out always, because we need to have a good relationship with explaining it to the, and not a 30-page or 20-page. I would say just a consent that is relatively easy to explain, that we can go over with our clients in detail. Volunteers.
Let's have the last word be from Kim Page, who, along with Andrea, ran the only phase 2 study for Hep C vaccine.
Yeah, I do want to note that in our previous trial, we did a lot of really interesting community engagement. And so it's not, you know, it's not something to point, and that was with people who inject drugs. Who is the community for a CHIM, you know, model is a little bit different, but we do have great experience and, you know, working relationships with folks who are at high risk, and experience writing consents and doing stuff like that. So don't discount the history that we have for multiple types of trials with these folks.
Yeah, great. Thanks for those points. Thanks, Kim. Thanks to everyone for the thoughtful comments and questions. There were a couple of really good questions posted online, and I think, Jake, they're for you. If you could, Jake Ebert, if you could wander over to the chat and address those. They're really good questions lingering there for you. In the meantime, thanks on behalf of all of us, thanks to you and your other volunteers for your sacrifice and for your altruism, and thanks to you for your talk, and to Charles, and I'll hand it back to Dr. Liang.
Well, thank you, David. It was a very lively and exciting discussion about the ethic and participant point of view. So let's move on to our second session. And it's really would be more related to many of the clinical questions that were posed regarding the DAA response and long-term consequences of cell-limited HCV infection. So the first presentation will be by Dr. Susanna Naglaa. She's the director of Duke Clinical Translational Institute, Vice Dean of Clinical and Translational Research, and Professor of Medicine at Duke University School of Medicine. Susanna, please proceed.
Great, thank you so much. And thanks to the organizers for giving me the opportunity to be here. I apologize that I was late in joining due to our annual CFAR meeting, but I've seen some great conversation already. Hopefully you can see the appropriate display of my screen. So I'm going to go ahead and kick this off. I know I have 10 minutes just to briefly intro some of the concepts regarding directly acting antivirals for the treatment of acute hepatitis C, given the discussion that we've heard here in terms of what is that risk-benefit balance when we think about asking volunteers to participate in the type of research that you all have been speaking about this morning. So I'm going to give a little bit of background, which is just for the entire audience, and thinking about, you know, when someone is exposed to the hepatitis C virus. and the way clinically that we diagnose someone or classify that infection in terms of the stage of the infection. And what we know for the majority of people who are exposed to hepatitis C, because we do not have a preventative vaccine, the majority go on to develop a chronic infection. We define acute infection as someone who is identified as being relatively early in their infection course within 6 months. And then early infection is 12 months. And I think, this used to be really relevant when we thought about therapies and treatment. And as we'll get to, I think it has become in many ways much less relevant from a treatment standpoint, because guidelines and clinical care have really moved along pretty significantly in the way that we manage patients with acute or early hepatitis C infection. I think everyone knows that hepatitis C is referred to as the silent epidemic. There's a reason for that, which is that the majority of people who develop acute, early, and go on to chronic infection never have symptoms that allow them to present to clinical care. That means the majority of them may have a mild viral syndrome that they think is any other virus, and that resolves, and that they go on to chronicity. They are not aware unless they see a provider or part of a screening program. So very few of these patients develop any sort of outward signs or symptoms of their acute infection, whether that be jaundice and or a true fulminant hepatitis. And I think that's one of the things to really think about here as we think about other viral hepatitis like hepatitis A or hepatitis B that really do have significant levels of patients presenting with severe liver injury. That is uncommon, extremely uncommon, in fact, for fulminant liver injury in hepatitis C. Really differentiates hepatitis C from a clinical perspective from these other viral hepatitis. You can see that because of direct-acting antiviral therapies, the guidelines, as I mentioned, have significantly changed. There was a time where there were recommendations that you actually wait to see if someone naturally clears this infection, which, as I mentioned, is an uncommon occurrence. And so now the recommendations from the primary guideline groups, whether in the United States, in North America, or outside of the US, including EASL guidelines and the APOSL guidelines, et cetera, is to treat people as soon as we identify them. Now, there is some difference, and we'll get into this a bit, in terms of what the recommendations are for therapy. So the AASLD-IDSA guidelines recommend treating someone right away, but treating them with the same length of therapy, the same therapy with direct-acting antivirals as you would someone who had chronic infection. So no differentiation of a treatment difference, whether you have acute, early, or chronic infection. The European guidelines do approach this a bit differently, which is a recommendation to use 8 weeks of one of two pan-genotypic regimens, sofosbuvir abatasphere or the glecaprevir preventosvir regimen, which I will refer to as GP moving forward because it's a whole lot easier to say. And we'll get into why that is. For soft-valve, this is actually a shortened course of therapy. For GP, this is the approved course of therapy for chronic infection, people who have naive or have never been treated or who have been treated with prior older regimens. So just want to summarize quickly, and really the landscape for this is to emphasize that it is really unbelievable where we are today with direct-acting antivirals. There really is largely only one population of patients that we take care of who do not have an approved access to DEA therapies, and those are pregnant people. But if you really think about it, beyond pregnant people, we have every patient who walks into my clinic would be able to and access DEA therapy. I'm not saying pregnant people can't, but that is not something that's approved currently or recommended per our guidelines. But what we do have is approval for patient populations, including children, and I really want to emphasize, and people who have gone through transplants of their organs, whether that be a heart, a liver, a kidney, and people who have decompensated liver disease. These are people who have very sick livers, and we have found that these regimens are very safe in people whose livers are not actively functioning at a level that would be considered normal or healthy for them. And we also now have what we call salvage regimens. And these are regimens where if someone has failed our first-line therapies, we can rescue them and treat them with highly active salvage regimens to ensure, frankly, that no one goes uncured. So I'm just going to summarize the data that we have here for acute hepatitis. And remember, we really do have very few large studies that are looking at people who have acute or early hepatitis C. And that is largely because Because if you look at the approvals for these drugs, these approvals have come in under chronic infection, and there frankly isn't a lot of reason for a company to pursue a shorter course of therapy, which is largely what we would look at in a clinical trial for an acute infection. So just to orient you to this slide, I'm largely looking at pan-genotypic regimens, so either the soft valve regimen or the GP regimen. So in blue are the soft valve regimens, and they have been studied. This regimen has been studied at both 8 weeks, which is a shortened course, as I mentioned, and recommended by the European guidelines, or 12 weeks, which is where it's approved in people who have chronic infection. And the dark and the light bars in blue are basically differentiating a clinical trial, what we call intent to treat population, which is would correlate less well to what I would consider a real world study versus the population that is the per protocol population, which is PP, which is basically a group who usually has at least reported that they've taken at least one dose of the trial medication, or potentially had reported significant adherence. And so what you can see here for the GP regimens is that we achieve 100% cure in people who say, I took the pill. And remember, these are people who actually who have acute infection and may actually be different than a population who would volunteer for a study because they are people who have recently been exposed to hepatitis C. And there may be social drivers of health that would be challenging for these patients to actually access care, et cetera. When we look at the GP regimen, you can see that these are in the orange and yellow bars. And again, orange is ITT and the GP regimen in yellow is the per protocol. This regimen, because it's approved at 8 weeks, has only really been studied at 4 and 6 weeks, because the idea behind acute infection is to see if we can use shortened courses of therapy for these patient populations. And you can see that even with a shortened course of therapy to 6 weeks, we can see cure rates of almost 96%. I do show you in the 8-week arm, which is where this drug is approved for 8 weeks, you get to a 98% cure rate. And these are people with chronic hepatitis C infection and cirrhosis and severe liver disease. This is very different than a population of volunteers who do not have underlying severe liver disease. So again, the point of this is we can cure near 100% of people, whether they have acute, chronic infection, cirrhosis, or other underlying comorbidities. So what about the safety of these regimens? I've already mentioned that these regimens are approved, and specifically the ladipasvir, sofosbuvir, and sofosbuvir velpatasvir regimens in people with decompensated liver disease. So they really are safe in people who have severe liver dysfunction. And this is true in someone who has acute hepatitis C, who has that rare occurrence of an acute liver injury. We can treat these patients, and we can treat them safely, and we can get them to a cure. And I think that's actually really important when you think about that risk benefit that we've been alluding to. So what if someone does fail that first line of therapy? What I'm going to tell you is that we can cure them. In fact, when I meet patients in my clinic, patients with severe liver disease, patients who have been previously treated, I reassure them that we will cure them. And what I tell them is, we will likely cure you on the first time. And if we don't cure you the first time, we will definitely cure you the second time. But we're not going to worry about that second time, because likely we won't need to have that conversation. Now, you can see that these are very small numbers that are summaries from the clinical trials that you saw on that previous slide of acute infection studies. And you can see there are very few numbers here because so few people fail to begin with. But for those people who do fail, you can see here that the cure rate is 100% on the second time around. And I'm going to summarize this here in a what I view as a good example of a population that has seen significant change in clinical care because of our level clinically of confidence in the efficacy and safety of these DAA therapies. So many of you may be aware that we now provide organ transplant to people without hepatitis C from a donor who has hepatitis C. So we are transplanting organs with hepatitis C infection in them, which will ultimately transmit that infection to the recipient at 100% rate, essentially. And we do that because we have such high confidence in our ability to cure them after the transplant. Now, you could argue the risk benefit is very different here, and I would agree with that. But remember, we're also treating these patients who are on the other end of that transplant significantly immunocompromised. Frequently, we'll also have other medical comorbidities, and we'll have significant drug-drug interactions that we manage And yet, as you can see here, the cure rate on the first round of treatment post-transplant is somewhere in the 96 to 100% range. And beyond that, if we retreat someone, it's 100%. And I actually give you an example here of our Duke experience, where out of 106 kidney transplants, 4 were not cured on the first round. These are very complex patients, and we cured those 4 with retreatment. And you could argue, why is that even publishable? But that just shows you how rare this experience is of needing to salvage someone in this setting. So to summarize, I think the considerations for this human infection model really is that we have treatment options. These are very different patient populations than what we're used to treating in clinical care, because they are volunteers and will not have some of the complications that we deal with in clinical practice. And I think these are some of the things that I wanted to put up just as maybe points of discussion. But ultimately, I do believe that these are patients, if they are transmitted the infection through studies, that we could ultimately rescue. And I will close there.
Oh, thank you, Susanna. It was a great summary of our experience in treating acute C with DAAs. So let's move on to our next presentation by David Thomas. Introduce him already, so he certainly needs no introduction. Please, David.
Thanks, Jake. I'm delighted to speak to this topic of the long-term consequences of a resolved infection. And I've actually sort of rephrased it, because I'm asking the question of what's the risk of 24 or fewer weeks of infection. That's really what we're talking about here, is what's the risk to persons who are volunteering for a CHIM. So for me, those risks boil down to liver complications and non-hepatic complications, and I'll talk about them in that order. So the hepatic complications of hepatitis C are, of course, cirrhosis, end-stage liver disease, and hepatocellular carcinoma. The non-hepatic complications, I'll group in the lymphoproliferative lymphoma kind of category, the vasculitis category, and then the metabolic vascular heart and diabetes kind of groupings. And for each of these, I'll be asking, is there any evidence that these kinds of complications occur more often in persons with spontaneous clearance, being what I consider to be perhaps the best example, the best model to answer the question of what the risk would be for someone in whom we allowed infection to go on for up to 24 weeks. So do they occur more often in persons with spontaneous clearance compared to persons who have persistent infection? And then kind of insinuating some information from those that have had the infection for a few decades and then received DAAs. So that those will be our sort of paradigms, if you will. Okay, so I'm going to disclaim right from the beginning that there are, of course, some limitations. These aren't randomized controlled trials. I'm trying to make inferences from secondary sources of information. There haven't been randomized trials with decades of follow-up to answer these questions with the right amount of persons. So there's also the issue of confounding here, right? The people that get these that are participating in challenge studies may differ in many regards from persons who acquire infection naturally, but in their age and their comorbid conditions and competing mortalities. There's also things that are different about people that spontaneously clear compared to those that have decades of persistent infection. And it's possible that those could alter some of these inferences. And then we're talking about things that take decades to occur and occur from other things other than hepatitis C, which that makes this all very tough. Not only do they occur from conditions other than hepatitis C, but they're conditions that we can't measure very well. Smoking, alcohol use are sort of high on the list. So we don't really know how much anyone's ever ingested alcohol. So it's difficult to control and to carefully answer these questions. So with those disclosures, I'm going to pretty much conclude that I could find no evidence of long-term complications. And I'll take you through some of the data that led to those conclusions. First, for the liver studies, and I'll start with our own work from Baltimore, where we initially characterized hepatitis C infections in persons that had antibodies, and we didn't know at the time whether they had spontaneous clearance or not. And we noticed that the ones that had that there was a group that we later found out had no RNA, who had normal ALTs, who 20 of whom happened to have liver biopsies done, and in retrospect had no RNA and had no fibrosis, and then literally no elevated liver enzymes in a group of 177 without viremia, but in whom spontaneous clearance had occurred. We have also done extensive elastography, just a snapshot of time of 930 with undetectable RNA, and compared to 6,578 with viremia, and the elastography results are considerably lower. Now they're not all what we would consider normal, because of course we have some with BMI, high BMI, and some with alcohol use, but they're clearly lower. And then to this other paradigm of the DAA response, once individuals receive DAAs, we see marked reductions in cirrhosis, marked reductions in those fiber scans over time, with literally 72% reduction in the prevalence of cirrhosis at the cohort level after adjusting for HIV, BMI, and age. Okay, mine is not moving anymore. Let's see if I can move it this way. Okay. Another cohort where we have spontaneous clearance results is that I'll show multiple times is the Taiwan Reveal cohort, 23,820 individuals, 30 to 65 years of age, enrolled from 1991 to 1992. So long-term cohort looking at mortality, cause-specific mortality, comparing those with anti-hepatitis C, with antibodies to hepatitis C, according to whether they're RNA positive or not. And you'll see a family of curves. I'll show you the first one here in cumulative mortality related to chronic liver disease and cirrhosis on the y-axis, time on the x. The little dotted lines are the RNA negatives, the solid of the RNA positives. No evidence of long-term cirrhosis or end-stage liver disease in those who had spontaneous clearance. Another valid control group are the German anti-D, and it's similar findings from the Irish anti-D. These are women who were accidentally inoculated with immune globulin contaminated with hepatitis C. In this study, 718 women who were hep C antibody positive, and 189 of them had spontaneous clearance. 2 of those had cirrhosis, but one was clearly from alcohol, and the other one had alcohol in the background as well. So these women, the authors concluded, spontaneously recovered and had no permanently cleared, were protected from liver fibrosis progression. So another form of negative information. So I show you those examples to reach the conclusion that I find no valid evidence that 24 or fewer weeks of viremia would lead to serious end-stage liver disease or cirrhosis or liver fibrosis accumulation. The other liver outcome, of course, is hepatocellular carcinoma, and there's some really intriguing in vitro and actually ex vivo data on liver epigenetic changes in liver that might have suggested that that hep C replication alone might lead to hepatocellular carcinoma. But kudos to Christine and Jordan and David and others who put this paper, this massive paper, into the CID supplement. I won't go over the details of it, but I'll just say that this is your one-stop shopping to answer this question. These are some of the papers that they highlight for spontaneous clearance. Notice in the third column, the incidence per 100 person years from some of these same cohorts I've already presented to you, the German anti-D women, the Irish anti-D women, that Reveal one I've already shown, and then a large VA cohort and a cohort from British Columbia. And in these cohorts, very low incidence of hepatocellular carcinoma to 0 in persons with had spontaneous clearance. So I found no conclusive evidence, despite the biologic plausibility presented by those intriguing histologic findings, that there would be any risk that we seriously need to consider. And here are those reveal data. I've summarized them in that table, but here are their Kaplan-Meier figures, cumulative mortality on the y-axis, reveal cohort, comparing those with spontaneous clearance in the little solid line, the solid gray line, persistent infection in the black line, and then in the dotted line, individuals that were antibody negative. And you can see just a slight increase in the antibody positives, but even these authors concluded that there was that could have been confounded and was unlikely to have been caused by the hepatitis C infection. Notice that in the first 10 years, there were no differences. So that's cirrhosis, that's hepatocellular carcinoma. This other category of non-extrahepatic, I'm going to highlight this paper for your one-stop shopping. Patrice Kukub has done most of the work on this topic and is a person whose opinion I respect. I asked him, because it's a little bit more complicated to get your head around these outcomes, if by email a couple of days ago, and he says if patients spontaneously clear their virus, they have no risk to develop such extrahepatic complications. That's his view. And some of the data that contribute to that would, for example, one of this reveal cohort that I've been showing from Taiwan, again, those with Hep C RNA in the black line have a higher mortality related to extrahepatic disease than those who are Hep C negative and Hep C antibody positive, RNA negative, and those lines overlap. The Irish immune globulin cohort, again, women infected at relatively young ages who had hepatitis C according to whether they were PCR positive or negative. And you can see that none of these persons infected at a young age developed vasculitis, whereas there were a considerable number in those that were Hep C RNA positive. The VA cohort has also done some massive studies using their ICD-9 coding, and again, substantially fewer instances of extrahepatic cryoglobulinemia, porphyria, lichen planus in those according to the RNA status. Here, looking at SVR compared to no SVR. So this is among persons with decades on average of infection after SVR compared to before SVR. So in conclusion, my summary, brief as it is that having reviewed a number of these papers, I really can't find any evidence of convincing long-term hepatic or extrahepatic consequences from that can be attributed to less than 24 weeks of HCV infection, and that includes the well-known complications of Hep C, hepatic and extrahepatic, and the forms of evidence being the spontaneous versus persistence, added confidence by examining after DAA cure, and the caveat that we really, of course, couldn't exclude very low risks of long-term complications, just as with other CHIM paradigms. So I'm going to end there, Jake, and hand it back to you.
Great. Thank you, David. It's a really comprehensive review of the long-term risks of cell-limited acute HCV infection. So I think we're going to move on to the second panel, which will be run by Jordan Feld. Jordan is a professor of medicine, interim director of Toronto Center for Liver Disease, and Phelan Chair in Translational Liver Research, Disease Research at the University of Toronto. So Jordan, please proceed with a panel discussion, which I very much will look forward to, because we have a lot of really world-class experts in hepatitis C to chime in on these topics.
Great, thanks, Jake, and thanks to Susanna and Dave for great presentations to introduce this panel, and great to have both of you on the panel. And in addition, we have some very illustrious colleagues who need little introduction, but I'll just mention their names and where they're signing in from. We have Graham Foster from Queen Mary University of London. We have Christine Su, who put together that great systematic review on the risk of liver cancer, who works in the liver diseases branch at the NIH. We have Jürgen Rockstroh from the University Hospital Bonn, and Nora Thoreau from the Keck School of Medicine at the University of Southern California. So great to have all of you. And maybe we'll just go back to the panel view. And I have a few questions that come out of the discussion. And I also would encourage any people who are viewing to put your questions in the Q&A, and be happy to share them with the panelists. And I like Graham's eagerness because he's got his hand up before I even ask a question. So I'm going to say, Graham, please give me your comment before I ask a question.
Thanks very much, Jay. I'm going to go to David, if I may, about the thorny issue of a post-viral fatigue syndrome. I think all of us who have published on hepatitis C-associated symptoms are aware people with infection do have a chronic fatigue syndrome that mostly abates when you clear the virus. But there are a cohort of people who have a persistent fatigue syndrome that may or may not be related to infection. My anecdotal experience is there are a group of people who are antibody positive, RNA negative, who do complain of chronic fatigue and the brain fog. Many of them associate it with infection. I'm not aware of any high quality research investigating it. It is mainly anecdotal, but there are certainly cohorts of patients whose fatigue coincides with infection. It seems to be particularly common in some countries. I have some exposure to Irish ladies infected in the anti-D cohort, many of whom who cleared and do seem to have a fairly persistent post-viral fatigue. So I'm keen to hear other colleagues' views on whether this is a real issue and how we should deal with it in a challenge trial.
Dave, do you want to start since the...
Yeah, I can. Graham, it's a great point. My final bullet where I said that I didn't feel like there were any risks different than other CHIMs was actually written thinking of that point of yours. In other words, any infectious disease can, of course, cause post-infectious fatigue, and so yeah, I mean, I do think it's possible that might occur. I had thought that the Irish women did do some, or the Irish investigators had done some formal testing, neuropsychiatric type testing, and concluded that the lack of viremia was associated with much lower depression and CNS. But you sounds like you have firsthand experience seeing them, so I certainly defer to you on that point.
I think on a population level, there does seem to be a dramatic improvement, but there's no question there's a small cohort of people who have persistent symptoms. So I think we need to factor that into the patient information. And I'm aware there's been quite a correspondence in the chat about how detailed we need to be, but my argument would be this is something we need to highlight.
I think it's a great point, Graham, to bring up. And I think one of the things that I know Christine and I spent a lot of time thinking about with others is this question of how do you actually answer these questions? It's very hard to sort of prove a negative, and particularly with things which are harder to measure, like fatigue, but even with very objective metrics of liver cancer, one of the things, and maybe Christine can comment on it, is this issue of how when people are tested, is that a confounding issue in and of itself? So are we really getting a true population of people who've been exposed and cleared versus a population of people who have a reason for testing? Christine?
Yeah, I think the challenge is that, we just have very limited data on I was looking for spontaneously clear cohorts, and we have very limited cohorts that kind of have a natural history, long-term health complications. Even with the, the Taiwan Reveal cohort, they, hepatitis B, core positivity was not accounted for, which can be a confounder in HCC outcomes. With the U.S. Veterans Affairs cohort, patients were tested for, at-risk testing. They were tested for liver risk factors, which led to the Hep C diagnosis. So a lot of patients had underlying, comorbid conditions that may have not been captured, like underdiagnosed MASLD or underdiagnosed alcohol, associated, liver disease. So I think it's hard because I think I think with the fatigue, a lot of the patients that have the fatigue have the chronic infections. And I think trying to extrapolate that to kind of apply to people who are treated in less than, 24 weeks has been difficult. And, with the spontaneously clear cohort, while majority of the patients clear in the first 6 months, but there have been some patients that cleared, beyond 6 months. So it's really hard to understand if this is truly reflective of the healthy population we would recruit for the CHIM. So.
Great. I'm going to go back to the discussion about treatment in acute hepatitis C. Thanks, Susanna, for that great overview about it. I can tell you when we talk about CHIM, the first question that anybody asks is, what about the 5% who don't respond? So you sort of showed us that seems unlikely in the acute setting. But I guess I just want to push you a little bit on that and ask you, how do we explain this and how confident can we be about curing everybody?
Yeah, I mean, I think the data does largely speak for itself, and you don't want to. I think you do have to make sure that folks understand that promising 100%, I think, can be difficult. But frankly, I do feel quite confident in saying that with the current DAs and options for potential rescue on a second round, that we can cure 100% of people. I think that the data that we have across the spectrum of people with hepatitis C says that's true. And I also think that the access to drugs is going to be different for this population than in people who have, as I mentioned earlier, drug-drug interactions and other complicating, potentially comorbidities, et cetera, that can sometimes limit how we treat folks, right? I mean, I think that was true in the transplant population. But I'm pretty confident based on the data and clinical experience that we can rescue any percent that fails the first time. I think 5% is high in this population. Honestly, I think we're really talking about the potential for 1 to 2% max of potentially needing retreatment, if any, if you honestly.
Jürgen?
Yeah, let me just comment on that question you raised, which I think Susanna already answered very nicely. I think we have to take into account that a lot of the studies in people with recently acquired hepatitis C were done in certain populations, including people with HIV co-infection and then drug users. And obviously they come with a lot of comorbidities. Remember, the clearance rate in HIV already is much lower. So there are special things. The hallmark of acute hepatitis C and HIV is a much higher viral load. And that explains why some people were struggling to respond. Remember that the failures we've seen in the acute hepatitis C trials mostly derive from people with very high viral load, but that is mostly related to people who have HIV and who come with a higher viral load. And if you look at the longer treatment durations or the standard, and I guess that's one of the reasons why AASLD comes with a recommendation to use the same dosages as in chronic infection, then actually the overall failure rate goes towards zero. So I think it's then more sort of this attempt of physicians to think that potentially because of the high cost of the drugs, we can treat shorter. Because remember, in the old days when we gave interferon, we got away with much shorter treatment durations and could spare a lot of the toxicity. And we always believed that there are different immune mechanisms and things would be much easier in acute hepatitis C, but that may not be true for shortening DAAs even further. And I think if we stick with the standard recommended dosages, then I think we should be fine. And the risk is really, really less than 1%, quite frankly.
Susanna, do you want to add to that?
Yeah, I was going to address, I see a comment in the chat on the 5% failure. I think this is again, I would really emphasize too, and I kind of put this in my summary slide that I blew right through, which is, I think in doing this, right, you want to make sure that we have learned through the trials for DAAs that there are novel variants, right, out there in terms of subgenotypes, et cetera, that have at times been, we've seen potentially more challenging to treat with pangenotypic regimen. So I think this is a controlled situation, right, where we can ensure that the viral strains that we are involving in these studies are in fact ones that we are highly confident and well experienced in treating and curing versus ones that we have discovered throughout the global trials.
So when we come to, I mean, we've been sort of talking about the DAAs fairly generically as treatment for this, and you did show us data from both the common pangenotypic regimens. Maybe I'll ask Nora, do you think there's a difference? Do you think there's a preference for one DAA regimen over another? Does it matter which genotype we're treating? Any thoughts?
I honestly think that for a treatment naive patient that, you know, the main differentiating with which one you choose is based on drug interactions. Quite honestly, that's usually the thing that drives you to choose one over another. And I think in a CHIM study, you're going to be thoughtful about ensuring that people are not on medications that would be a contraindication, for example, to using one of these regimens. For example, we all know that some anticonvulsants are actually very challenging to use with either of our DAA options. So, you know, just being thoughtful about those kinds of things, I think would mean that we'd be fine in terms of, you know, selecting one or the other. I think the key thing is having access, as has been highlighted. So I personally think they both are strong candidates as the first line treatment, and I wouldn't choose one over the other except based on DAAs. And that's true for genotype 3, which you might be getting at a little bit, but I think actually as initial therapy, I still am perfectly fine in terms of thinking about using either of these as first line treatment.
Yeah, and I certainly don't want to push on that point. I agree with you entirely. And Susanna mentioned about genotype in her comments. So I guess would we think differently about the different genotypes? It sounds like most of us, I think, would be comfortable saying it doesn't matter with, remember, this is a person this is an acute infection and someone without liver disease and a first round treatment. What about, I guess I'll just throw it out there, but any concerns about using a protease inhibitor containing regimen in someone with more severe acute infection? I see some head shaking, but Graham, I'll put you on the spot. What do you think?
Thanks. No, I don't. I wouldn't be too concerned about a protease. I would just push back a little bit on the genotype 3 question. I think most of the genotype 3 we see is 3A, which is probably non-problematic. The 3B has got associated issues with it. Some of the genotype 4s have got concerns about sofosbuvir usage. I think one of the slightly broader questions here is, what is our target audience? And are we looking for a pan-genotypic vaccine? And I think Michael raised in the earlier session this rather important point of feed through. Are we looking to develop a vaccine for US? Are we thinking about a Pakistan type vaccine or a genotype 6 vaccine for Vietnam? Or do we need a pan-genotypic? And I think that needs a little bit of thought when we consider the strains that we're going to use. I think if we're going to Pakistan, where 3B is 15% of the population with a slightly lower sensitivity to sofosbuvir, you might want to reconsider your approach to using a blind, any drug combo will do. So I think there does need just to be a little bit of thought as to our target audience when we think about genotypes and therapy.
Jake?
Yeah, I mean, just getting back to the genotype, I think for the CHIM study, it's somewhat of a moot academic point genotype, because we'll be selecting a particular inoculum that has been shown to be responsive to DAA, right? That's one of the, obviously, the criteria we will use. So it's probably not as relevant, but certainly we would need to be very careful in choosing the correct inoculum, which will be discussed further in the next panel as well.
Yeah, it's a great point. Ray Chung's going to talk about the selection of the inoculum. And when I talk about the clinical trial design, I will highlight that point that any inoculum we use will be an inoculum that has been shown to be sensitive to the DAAs we are going to use in the trial. And really what I was asking a bit more generally was whether there would be any preference for one regimen broadly over another, which it sounds like probably not. That would be more related to the safety of the drugs in the context of potentially a more severe acute hepatitis. But it sounds like most people are pretty comfortable with a protease inhibitor in that setting, and I would tend to agree. Nora, I see your hand.
Well, I was going to just disagree a little bit about the protease inhibitor. If the bilirubin is elevated, I would move away from a PI inclusive regimen. So I think if the bilirubin, is over 3, you have somebody who's really manifesting the more severe end of the spectrum where the bilirubin is rising, and now they're moving to being, say, somebody who's got a Child-Pugh score that's now 7 because their bilirubin is elevated or their INR is up. I would not use a PI inclusive regimen there. I would move towards a PI, one without a PI, just in terms of the information we have around safety. It's not a huge point, but I think we have choices, and I would move towards one that did not include a protease inhibitor if they're manifesting that kind of severity.
Great, thanks. And that was sort of the point that I was trying to raise with the acute hepatitis. And I think there really are not a lot of data in this setting because, as Susanna pointed out, very severe acute hepatitis C is fortunately rare. But it is also reassuring that in the setting of even fibrosing cholestatic hepatitis, in the setting of liver or other organ transplants, that therapy is effective even with that very severe form of acute hepatitis C, and people have tended to use sofosbuvir NS5A based regimens in that setting. To be honest, I'm not aware of whether protease inhibitors have been used in that setting, but that's a reasonable consideration. So moving back to this complications question, because this is the one that really, I think, is the thing that keeps all of us up a little bit at night. Are we going to be, I think most of us are pretty comfortable we can cure the people if they don't clear on their own. But I think we are worried, is there something we're doing long term? And I think one of the questions that comes back to this question of duration is whether we think the duration of the acute infection, 1 month, 3 months, 6 months, does that affect the likelihood of developing any of these chronic sequelae if they are to develop? Maybe, Graham, since you brought up the question about post-fatigue, I'll turn it to you to start, but I'd be interested to hear what others think as well.
I suspect that it will be duration of infection related. And I say that without any good data, but it just seems to me intuitively, if you have a brief infection of 3-4 weeks that's aborted, rather than if you started to get your immune response, your interferon response engaged for a prolonged period of time. So my suspicion would be that it will be duration related. I've no evidence to support that, but it just feels more sensible to be conservative. So I would be more comfortable aborting these infections at the earliest stage. I think what we're looking at surely is a sterilizing immunity. So you give people a slug of virus, they don't get any viremia at all. So that question can be answered, I think, with a fairly brief exposure. And my view would be that's more likely to reduce the risk of off-target effects.
We'll come to that discussion of sterilizing versus non-sterilizing immunity, because and there's definitely some differing opinions on that. We will discuss that, I think, at some length later in the session. So I'm going to I'm less optimistic that we're going to achieve sterilizing immunity, but I'll turn it to Nora to weigh in on this question of duration.
Well, I'm going to sort of take a slightly, I think this idea that there may be epigenetic changes that occur in the liver related to kind of more prolonged infection is something that we should be a little bit thoughtful about. And I appreciate the data looking in particular at HCC, which probably is kind of the major concern I think that most of us would have, if whether there's any, if it confers any increased risk. The thing I'm a little bit just aware of is that the patients that we're going to propose to infect in 2023 or 2024 are very different from the cohorts 2 decades ago, which is the data you're looking at. The presence of especially mazzled in the population, the fact that you're going to have individuals that probably have some degree of liver, they may have liver injury even related to fat in the liver. We just have to be aware of that. And is that an environment in which when you have Hep C infection that the outcomes are going to be in some way modified or changed? So just thinking about the patients that we currently have, like, the frequency, especially of metabolic dysfunction associated liver disease these days is important, I think. So I would just, I think that that's my concern. And so longer for me, I also would be, like Graham, more enthusiastic about a shorter exposure with no data, but really this idea that, longer viremia, longer the sort of ability to have kind of this, having immune response within the liver, epigenetic changes, I'd just be concerned about long-term consequences. We just should be thoughtful about that piece, I think.
Absolutely. And I'm going to talk a little bit about that in my discussion about clinical trial design and the rationale of earlier versus later treatment. So we'll save some of that discussion for later, but Jürgen.
Yeah, I just want to follow up one thought on the fatigue issue. I mean, if you think of COVID, because we now have so much research going into long COVID syndromes, there the appearance of the syndrome is very much correlated with presence of viremia, and if viremia goes down early, then it's less likely. So I think there's, at least from other viral infections, some evidence which supports that duration of viremia may matter. And if you believe in these concepts of, first or second hit of liver damage, I think if there's pre-existence changes in the liver, that may have an impact also on duration of infection if there's already something pre-existing which may worsen then. Eventually, I would, though, argue that in the setting of a new concept which is being developed, you're going to choose patients who are liver healthy. So I think that risk can probably be eliminated, at least largely, by inclusion of your first volunteers.
Great, thanks, Jürgen. Dave?
Yeah, just I wanted to make clear that I agree that epigenetic changes are important, but I believe they probably occur with every infection that you get. And the available data suggests they occur fairly quickly. And I didn't get into these details. I actually hid the slide so I could get done in time. But the ones that were in the gastro paper, as we discussed in the chat, were principally 60-some% of it was explaining fibrosis, was an indicator of F3, F4 fibrosis, not an indicator of Hep C infection. So I think I'm worried about that, and I'm worried about it for like the post-infectious fatigue. I'm worried about it for other reasons as well. I don't think it's unique to this CHIM. I think all CHIMs should have to, all CHIMs have that issue. And anytime you give someone an infection, it's a real concern. I think when you think about the time scale, it's worth thinking about it is 6 months. 6 months is double 3 months, but 6 months compared to 3 months compared to 30 years, and so I think it depends. Most antibody specificities have expressed themselves by 3 months and 6 months. So I don't know. I really don't know, and I wonder if it's more subjective. And I would come back to your later point. I know you're going to make this, so I love it. If I would rather have 6 if it makes a study valid, because if we theoretically feel like maybe there's some increased risk, but we invalidate the whole study by cutting it at 3, then that doesn't do anyone any good. So those are my thoughts.
Yeah, and I guess just to add a little comment to the epigenetic changes, because I will tell you, we spent a lot of time poring over those data. And I think one thing that's really interesting is that you really do see in the in vitro models, you see the epigenetic changes 100% of the time. And in people with chronic Hep C infection, when you biopsy them, you see the epigenetic changes 100% of the time. What you see is that the people who developed cancer in those cohorts were exclusively people with cirrhosis. So my take on it is that this is a phenomenon that happens with Hep C infection, but that you don't develop liver cancer unless you develop advanced liver disease. And at least in this setting, and there are no data on whether these epigenetic changes occur with acute infection or acute resolving infection, and whether they disappear over time, to know the real answer to that question. But I think it's important to really highlight the fact that cancer almost never occurs in the absence of fibrosis, and most commonly occurs only in people with cirrhosis, even if it's after cure. So I guess this brings up another question, which is how long do we have to follow people after a CHIM? So we're talking about these long-term consequences. Some of them might happen decades after we expose people. So Christine, what's a reasonable time frame to be thinking about asking someone to keep coming back for annual evaluation?
I mean, based on sort of, the meta-analysis we did and kind of looking at these cohorts, it seems like the, HCC risk is, minimal, if not negligible, and very low. And, the rates are not at rates where there's an indication for HCC screening. So, I mean, I guess if you want to be cautious, I would like to follow them as long as possible, because I think risk is not defined by one time point. Risk is a trajectory, right? And so, we take these healthy volunteers, and if all of a sudden, one healthy volunteer is drinking heavily or has gained a lot of weight or developed diabetes, that may change their risk of HCC. And so, I certainly, I don't have a good answer, but I guess I would follow annually until somebody tells me I have to stop following them. So for me, I feel like risk is not necessarily defined by baseline demographics. So.
I think that's a great point. And I think all of us feel that we certainly need to follow people long term. And I guess the question is, what is long term? Is 10 years? It seems like a long time for people, but when we're talking about some of these very late complications, we really might be thinking about 20 or 30 years, and whether that's practical to ask somebody to commit to that. I think that's a lot to ask of them. But on the flip side, there is potential benefit for them of early diagnosis or identification if problems were to occur. So there may be some benefits of that. And I see a comment in the chat that I think people feel like probably 10 years is probably the minimum, but whether we need to go longer is a question. So I think we are. I think our time is about up. I'm looking to Jake to see if there's anything.
Well, certainly we can take an earlier break if people have enough discussion. So I'll leave it up to the panel and people in the audience. I think there are a couple questions in the Q&A. You may want to just look at
yeah, and I think some of these questions probably relate a little bit to some of the other topics, but we have a great panel here, so maybe I'll ask them what they think about them. And I think this is an interesting risk question, because we don't necessarily think of it as a risk in terms of the disease, but what about any risk of stigmatizing or insurance discrimination for someone who tests Hep C antibody positive after participation in a CHIM? Susanna, what do you think?
Yeah, you actually saw that question, and I hope there's someone here who knows whether or not a HCV positive antibody is considered a pre-existing condition in the United States. My general understanding is that it's not, but someone may know better than I do, frankly.
Ellie, I see you. Okay.
Yeah, I do have a bit of experience of this through Hep C vaccines that we gave to patients, and they seroconverted and developed. Sorry, we gave them to healthy people, and they seroconverted, and they developed Hep C antibodies as a result of vaccination. And there are long-term consequences of that. For example, you can't be a blood donor in the UK if you're Hep C antibody positive. So. And the same thing would occur for these healthy people in a CHIM study. They would be excluded from future altruistic kind of endeavors, I think. So I think that does have to be considered, and it would be part of the informed consent.
And I see that Mark Solkowski's added in the chat here that people, at least in some settings, there may be screening for Hep C antibody by insurance companies. If they test positive, then it's beholden on the person to prove that they're negative, which is just an added barrier. So, it's certainly something that needs to be in a consent form. And I guess a question beyond that is whether it poses any other risk. Andrea, I see you're here.
Yeah, I suspect Ellie did this too, but we actually provided a card to vaccine participants saying that they had potentially falsely positive HCV antibody tests as a result of participating in the trial, and provided a way to contact us for questions. Complicating our trial, which was in people actively injecting drugs, they could actually have hepatitis C virus infection. So we also educated participants and encourage them to have their providers call us, because HCV RNA would be the appropriate way to screen them for infection. We did not want people to not get tested for hepatitis C after leaving the trial, but in people at no risk for Hep C, that card is probably particularly important.
And it's a little bit different, obviously, in this setting, because it's not a false positive, it's a true positive. They were actively infected, and obviously the blood donation issue is really a little bit of a question about whether there's any long-term risk of transmission in someone who's PCR and NAT negative, which is why people have been excluded to date. Whether that will ever change is a question maybe for Harvey Alter, but in the current setting, it probably will continue to be an exclusion. So it is something to warn people. And for sure, this point about that they need to know that whether they've been vaccinated, not vaccinated, or otherwise, that they could still be reinfected. So they shouldn't assume that anybody positive is of no concern. Andrea, another comment?
No, I left my hand up, but I will say that I agree. And in talking to insurance providers, they have said that they assess risk differently because people who are antibody positive, it's a marker for higher risk behavior. I think that's very prejudicial, but that would not be true of someone participating in a vaccine trial. And that's my experience with insurance providers who've said, well, we view that as a marker of risk behavior that may change, for example, life insurance prospects. That I'm not sure it's fair, but it is something that has been brought up when I've challenged insurance providers about risk in someone who was treated.
Yeah, and I think that sort of gets to the point about stigma is real, and we shouldn't underestimate that, and people need to be advised of that if they are going to participate in something like this, that they may receive some stigma related to that, and hopefully that is changing and will continue to change, but it has not disappeared yet. Jake?
Yeah, I was just going to follow up on that question. And unfortunately, we don't have a set of insurance policy experts on the panel. I think in general, pre-existing condition cannot be considered in a lot of these insurance, especially national health insurance or other things that you associate with your company or Medicare. And so I think this is really more and more interest in not to consider these kind of pre-existing conditions. Actually, in some states, it may be illegal to do that, to exclude somebody because of pre-existing conditions. So I think that thing is going to change, but I certainly agree that provide some kind of oversight in terms of this person was involved in a vaccine trial, therefore they have antibody is very reasonable and easy to do. So I think we just have to see how that play out. So.
Okay, and I'm going to give Harvey Alter the last word on the on this.
Nothing insightful, but I didn't consider that possibility because they would exclude people from blood donation right now. That would require FDA changing the rules on donation. A lot of people are very devoted to blood donation, so for some people it would be a real negative. I don't think it's a big point, but especially with the small numbers you're going to enroll. But it is another consideration.
And I think it's important in the population that are going to volunteer to participate in a CHIM. On balance, this is a group that are probably more likely to be blood donors. They're more likely to be sort of health activity. So I think it is certainly something that needs to be in a consent form, and people need to understand that. And whether there ever will be a change in the policy around blood donation is obviously questionable, but probably not likely in the short term. So I think certainly a relevant point to have raised. So I think we've covered a lot of ground in this panel with some great talks and some good discussion. And I think it's good to be sort of mindful of some of the uncertainty around the long-term risks, but confident that the risks are low, albeit they may not be zero, and thinking about how we inform people about those risks and then how we follow them to ensure that we keep their safety top of mind will be things we have to think about carefully. So with that, I'm going to turn it over to Jake. Thank the panel members for all the great points and discussion, and move on with the session.
Again, thank you very much. And this is really a great morning we have, and I think we'll go ahead and take a break. So let's try to be back 12:40, so 20 minutes to 1, at least Eastern time. I know that may be different for European. So we'll come back and proceed with our last sessions, in which I think a lot of these maybe very similar question would be addressed as well. So everybody have a great break. We'll see you in a bit. Bye-bye. Well, hello, everyone. Welcome back. And we are going to our last session, perhaps a very important session in trying to realize CHIM into a true clinical trial. Before I do that, I forgot to mention that one of our speakers and panelists, that today is actually his birthday. That's David Thomas. So we will ask you how old you are, or what else you doing? I appreciate you doing this despite on your birthday. So happy birthday.
Thank you.
Happy birthday.
Happy birthday.
Mine was yesterday, 2 days ago.
Happy birthday.
Turning 50, Bobby.
I hit 52, actually.
Okay.
Happy birthday to both of you.
All right, well, let's go ahead with the next speaker. I think it's Dr. Raymond Chung, who's the Director of Hepatology in the Liver Center, Vice Chair of Gastroenterology and the Kevin Polly Maroney Research Scholar at the Massachusetts General Hospital, Professor of Medicine at Harvard Medical School. Ray?
Thank you so much, Jake. And let me see if I can share my screen here. Can you all see that?
You'll just want to put it in presentation mode. Perfect.
Can you see it now?
Looks great.
Okay, great, terrific. So thank you, Jake, and thank you to the organizers for asking me to represent this group, which was a considerable and formidable group, putting together the section for the CID supplement you referenced earlier, Jake, on the challenge inoculum or selection of challenge inoculum for an HCV CHIM trial. So just as a way by way of backdrop, it's certainly critical that we select an appropriate infectious inoculum. It's the cornerstone of a controlled human infection model. It's got to be reproducible, reliably infectious, and of course, above it all, safe. But it also needs to be relevant to human hepatitis C infection. It should, in theory, recapitulate chronicity. It should be a representative genotype. It should harbor the potential for quasi-species diversity, and of course, it should be infectious. So I'll just divide this short talk into several sections: the source of the inoculum, safety benchmarks, the characterization of that challenge inoculum, some discussion about viral genotype selection, and what an initial inoculation study to generate an acute inoculum might look like. And that will segue us nicely into the clinical trial design that George will get into in the next talk. So first, the source of the challenge inoculum. Well, there are a variety of potential sources, including, of course, the best characterized animal model, the chimpanzee, which, of course, is the only robust immune competent model for hepatitis C infection, capable of generating sequence diversity. But unfortunately, between the moratorium on chimp research and as well as safety concerns regarding potential passage of unknown pathogens, the chimpanzee is no longer a viable option as a source of an inoculum. There are, in fact, small animal models, humanized mouse models, which harbor human transplanted hepatocytes that in turn can be infected with hepatitis C and can generate infectious HCV. However, these are immunodeficient mice by design, and as with any small animal model, there could be unknown animal-derived pathogens. So this presents its own set of concerns for use in a CHIM model. Cell culture-derived virus is another potential source. The work of many on this panel have demonstrated that we can, in fact, take tissue culture adapted naturally occurring hepatitis C strains, such as the very well characterized JFH1 strain, and render it infectious in tissue culture. This is, however, a genotype 2A strain and derived from a patient with fulminant hepatitis in Japan. So there are some theoretical concerns there. Moreover, these were replicated in an adapted hepatoma cell line, and so are not compatible with GMP practices. Furthermore, there's no proof of replication in humans demonstrated for virus produced in cell culture, and is therefore not a practical choice for vaccine protection trials. So I've laid out some of the non-starters or less favorable sources. We come back, of course, to human infectious serum. which of course recapitulates all of the essential dimensions of human infection. It captures quasi-species diversity, and of course is known to be infectious. We have successfully, of course, been able to develop human-derived blood products, and this is a widely accepted practice with excellent safety profiles. So in theory, human serum may be the most attractive of the choices for a challenge inoculum. In terms of the nature of the infection from a human source, we have two stark or largely divisible choices between acute and chronic infections. Clearly, chronic infections are much more commonly available, and acute inocula are certainly far less common. They are, as we've discussed in previous talks, accompanied by considerably higher viral loads with much higher infectivity, and acute inocula have been demonstrated to be up to 1,000-fold or even higher in terms of their ability to be infectious in either animal models or tissue culture. The paradigm strain was that, of course, that Dr. Alter initially characterized, and that's the H77 strain, the genotype 1A strain that has been the source of so much insight about hepatitis C subsequently. It was an acute inoculum at high infectivity, very well characterized, and it's still, even after all these years, ample volume in storage. But it was stored under, at the time, not in the present day, clinically approved conditions. So if one were to pursue that, it would require recharacterization, including, of course, assessment of sensitivity to current directly acting antivirals. The chronic phase infection, of course, far more representative of most transmitted infections, is very common, and we can certainly find plenty of sources of chronic infection. However, what is absolutely clear is that infectivity of chronic inocula drops considerably, and that's likely due to antibody binding and immune complex formation, so that the inoculum volume required for a chronic source would no doubt increase, and with it would go the risk of adventitious unmeasured infectious agents or contaminants. It was established, again, using the H77 model in chimpanzees, that as few as 100 in immune complex free or free HCV RNAs are capable of establishing infection. Subsequent studies have used something more than that, about 3,000 RNAs in most studies that have proved over the years to be reliably infectious in both chimpanzees and humanized mouse models. So if we were to take an acute serum with harboring very high copy number, we could, in theory, yield up to thousands of infectious doses. And I wanted to just draw out that contrast, and this is a table that was generously provided by Jordan, and I've adapted it for this presentation, that if one were to take an acute versus a chronic source, and again, with the essential ask that we have 3,000 uncomplexed HCV RNA genomes, or free genomes, we'd start with an already higher viral load in the acute inoculum, 10 million, say, versus 1 million from a person with chronic HCV, translating out to about 25 million RNAs, or 2.5 million RNAs, respectively. However, if one were to look at free RNA genomes, that would largely be the case for every one of those genome copies in the acute scenario, whereas antibody binding and immune complex formation would render far fewer of the chronic samples truly free as HCV RNAs. And so one using an estimate of a 3-log reduction only yields 2,500 genome copies per milliliter, resulting then in nearly 10,000 infectious doses per milliliter in the acute inoculum, as compared to about 1 infectious dose per milliliter in a chronic inoculum, thereby increasing the plasma requirement per infectious dose dramatically from about 0.12 microliter to just over 1 milliliter. And if we were to take plasmapheresis and take donors, we could get, say, for 750 milliliter plasmapheresis, over 6 million infectious doses as compared to 600 in the chronic scenario. So the acute scenario certainly appears to be far more attractive. What about safety considerations? Well, the inoculum candidate would have to conform to all transfusion and blood product safety requirements, collected an approved blood bank with approved equipment, screened by standard tests, ABO blood testing, serologic and nucleic acid testing for other bloodborne pathogens such as HIV and hepatitis B, and processed and stored under GLP and GMP conditions. The donors should meet criteria for any blood product donation, and as we've discussed earlier, should be in very good health, with absolute plans for a DAA treatment after donation with clear-cut documentation of cure in those donors. We should exclude those who developed acute liver failure, fulminant hepatitis, and certainly those with more advanced forms of liver disease. We should, the attractive idea of taking someone post-organ transplant, in which even higher levels of viral RNA might be present with low concentrations of immune complexes, may be counterbalanced by the coexistence of occult pathogens in that group of patients. As far as blood type is concerned, the AB blood type would be preferred insofar as it is a universal plasma donor. But if we cannot identify just such a person, again, great pains to minimize inoculum size would be important. For characterization of the challenge inoculum, the primary goal is to establish infectivity and to target 100% infection in CHIM volunteers in order to maximize power and ethically acceptable study design. We would want to start with quantification of antibody-free HCV RNA, tested in cell culture and humanized liver mouse model to establish an infectivity measured in the form of MID50s. We'd want to sequence that inoculum and be sure that there were no significant resistance-associated substitutions. Once the inoculum was identified, we could then go back and evaluate the sensitivity of that inoculum to neutralizing antibodies and antisera from previously vaccinated mice or humans. What about genotype? Well, it's ideal to have a genotype-specific challenge inoculum for each of the 6 major genotypes. This might prove impractical, at least for initial CHIM studies. However, it may be useful to have more than one. So somewhere in the middle, I think 2 genotypes may be a reasonable choice, and the 2 most perhaps attractive genotypes would be genotypes 1 and 3, which are clearly most common among those at high risk for HCV transmission, namely persons who inject drugs, responsible for nearly 90% of infections between those 2 genotypes. Even though we have heard earlier that there might be slightly diminished DAA responses among genotype 3 patients, again, we would take great pains to confirm cure of the donor with DAAs before advancing with that specific genotype 3 inoculum. And we could do this as a parallel processing exercise in which collection and preclinical characterization of multiple genotype inocula could be pursued, again, simultaneously. So how would we start? And this would be, again, a segue to the ultimate vaccine study. And we would start with a first stage study. And this would be a small scale inoculation study. This would essentially establish a dose that would lead to reproducible infection. If we were starting with an infectious serum study, it may be reasonable to start with that 3,000 RNA genome dose in the first volunteer. And if no infection were to result, we could ratchet up by increasing the dose, perhaps by a log, as a dose-finding exercise. Those who do establish infection would be followed, and again, we've had that discussion this morning, for 12 to 24 weeks, then apply curative DAAs. Samples from this initial acute inoculum study in these volunteers could be characterized further and then used subsequently as the source of challenge inoculum for the more definitive stage studies, the stage 2 and 3 studies you'll hear more about in just a moment. These participants could also be controls for observation, spontaneous clearance, and vaccination studies, again described subsequently. One consideration as an alternative to infectious serum I'd just like to present would be the notion of taking molecular clones of infectious HCV that have been established for actually a number of different genotypes and subtypes, such as those displayed here, whose course is comparable to infection with HCV-infected serum, as again has been demonstrated amply, and can be successfully passaged to other chimps and humanized mice. This may represent an even safer approach than the human source inoculum, particularly as we consider regulatory constraints. And perhaps, again, the only limitation to a clonal source of an infectious RNA in this case would be, does it recapitulate the diversity of natural infection? And I think to that, the reasonable counter would be that as we have looked at transmission of acute infection, in general, despite the quasi-species existence of the donor source, we have seen bottlenecks in transmission, with generally speaking, a single quasi-species emerging as the dominant in the infected recipient. The other challenge, I think, for an infectious RNA would be, can it be delivered without direct intrahepatic inoculation, such as was performed in the original chimpanzee studies of infectious cDNAs. So to this point, there are actually, I think, advances in therapeutic technologies that have allowed us to deliver peripherally RNAs into patients. And this has been aided and abetted by the use of cationic lipid nanoparticles, which have been well established and tolerated in clinical trials, including trials of not just vaccines, but gene therapy, RNAi, and antisense approaches. And RNA lipid nanoparticles could be produced in a GLP-GMP facility for human application. And the great majority of these would naturally target after intravenous inoculation to the liver. So this may represent an attractive approach to generate challenge inocula in humans. So to close, this is what a stage 1 study might look like in order to establish that challenge inoculum using a source, again, whether an acute patient or chronic patient, or potentially even an infectious RNA tagged to a lipid nanoparticle. This would, again, corresponding perhaps to genotype 1 and genotype 3, in a patient who had been subsequently cured and documented for cure. 3,000 of these genomes would then be inoculated into the initial recipient, and we would start with 1 dose per genotype. These patients would be tracked frequently pre-viremia, and then once viremia was documented, again, tracked serially until a titer developed in excess of 1 million IUs per mil, after which plasmapheresis could be performed to collect large volumes of this inoculum, which would then, of course, be a potential source for future CHIM studies, which we'll hear more about. At the point of either seroconversion, the development of severe hepatitis, or after the second plasmapheresis has been completed, these patients would then be offered curative therapy with glucapavir and probenespir, just as an example. So with that, I'll just conclude by saying that selection of a suitable challenge inoculum is absolutely cardinal for successful CHIM. It must be reproducible, reliably infectious, safe, and relevant. It would appear that human infectious serum from a high titer acute HCV infection may be the most attractive source. We should establish infectivity in animal models and then plan initial dose finding inoculation studies. The stage 1 study I just described. Infectious clonal RNA may be an even safer and more tractable alternative to generate an acute inoculum. And we should strive for several genotypes, and I would make the case that genotypes 1 and 3 may be the most attractive among those. And again, the hope here is that this ideal challenge inoculum will put us in the best possible position for success of a candidate vaccine trial. So thank you for your attention.
All right, Ray. Well, thank you very much. I certainly could not have done it better, even though you referenced my namesake sake as that particular CID article. So we can move on to the next presentation. Ray, maybe you can unshare. Great. All right, well, next presentation is by Jordan Feld. I introduced him already. So without further ado, take it on, Jordan.
Jordan, I think you might be sharing the wrong screen. We're seeing the presenter notes. I think you also might be on mute. Jordan, do you want to come off mute, please?
Yeah, sorry about that. I'm just, do you have the right screen up now?
Yes, you just have to put in a slideshow.
Yeah, okay, sorry about that. I did sometimes, I have two screens and sometimes it shows the wrong one. There we go. Good? Or you're still seeing the presenter?
Yep, that's good, yep.
Okay, apologies for the delay. Thanks, Jake, for the introduction. Thanks to everybody for the participation in what's so far been a great session. And so what I'm going to talk about now is really thinking about clinical trial design, and I'm going to summarize some of the discussions that we had that we put in the CID supplement, and I've acknowledged the co-authors on that article below. So I'll talk about really the strategy here, vaccine endpoints. We've talked about some of these things already today, but I'm going to highlight where we at least felt that the consensus coming out of these workshops that we've had, as well as discussions leading up to now, point us in the direction. So the endpoints, the population, the actual trial design, what we're going to do in terms of the indications for treatment, a fair bit about safety considerations, and then some futility criteria. So when we think about this, it's important to really think about what are the preferred endpoints for any vaccine study. And it was actually discussed in the panel discussion earlier that Graham Foster said, well, of course, you're going to aim for sterilizing immunity. And I think all of us would love to see sterilizing immunity. That's the idea that you prevent initial infection. There's clear benefits to that. There's no acute infection. There's no transmission after acute infection. And the best thing about it for a CHIM is it's actually very easy to assess. The problem with it is it may be a very high bar. The alternative would be to prevent chronic infection. So in this concept, you would aim to effectively increase the spontaneous clearance rate in people who receive the vaccine. And the benefit here is, although you don't prevent acute infection, you still prevent the long-term complications of hepatitis C, particularly of advanced liver disease and of non-liver related complications and liver cancer. An alternative approach in theory for some pathogens is to try to attenuate the infection. So you have reduced severity of infection, sort of like what's been seen with COVID-19. This clearly isn't feasible in a Hep C CHIM model because the complications happen over years or decades of infection. So if we sort of think about this concept of sterilizing immunity versus prevention of chronicity, there's no question that this sterilizing immunity would be attractive. The problem with it is that it's been difficult to achieve. So in this setting, someone has protective antibodies and presumably and potentially T cell responses. They get an infection and they never develop viremia. This has been achieved, I'll show you, with homologous challenge in chimps, where chimps have been challenged with a virus that was used to develop the vaccine, but it has not been seen with either reinfection or heterologous challenge in chimps, and I'll show you these data in a second, whereas prevention of chronicity, this idea that somebody develops an acute infection but then spontaneously clears it like we see in natural infection, but just at a more frequent incidence in people who are vaccinated. This has been achieved with heterologous challenge in chimps, and it's also been achieved with reinfection, where we know that people develop at least partial protective immunity with lower peak viremia and more rapid clearance on repeated infections. So prevention of chronicity would avoid the major consequences of Hep C, and although sterilizing immunity might be the goal, the problem is it may miss an imperfect but still effective vaccine. And I think The feeling has generally been that the bar really of setting for only sterilizing immunity might be too high a bar, and that really the primary aim of the initial vaccine trials, including CHIM, should really be prevention of chronic infection across all of the common 6 genotypes. So just to show you some of the data, these go back for a long time for Michael Houghton's team, looking at what happens when you give a chimpanzee an infection. You can see that with chronic infection, you get viremia, an ALT elevation, and then persistence of viremia. Now, when chimps were vaccinated with the same virus that was used to make the vaccine, you could see that it was possible to develop sterilizing immunity where the chimp never became viremic despite being inoculated. And the alternative here is seeing a chimp that becomes viremic. So you can see they develop positive viremia shown here, but then clear that infection. And when you look at the collection of data from Michael Houghton's team and their studies, you can see when you do a homologous challenge that if you give, in this example, 12 chimps this vaccine candidate, an E1, E2 vaccine candidate, you can see that 7 of the 12 developed acute infection, so 5 actually had prevention or sterilizing immunity, but you can see that the benefit was greater in terms of chronic infection, and this was this difference was much more dramatic when you gave a heterologous challenge. So you give a different virus, in this case, not the virus used to generate the vaccine. And then you see that all of the chimps developed acute infection, but still only a small proportion of them developed chronic infection. And when you look at this overall, you get a high clearance rate in the vaccinated chimps, that only 16% developed chronic infection compared to 63% of the unimmunized. And so this really sort of makes the case that because sterilizing immunity is achieved only with virus used for the vaccine design or homologous challenge, it's probably a pretty high bar for us in our first efforts to make a vaccine. And prevention of chronic infection is still a major benefit, and I think should be the target, at least initially. We'd certainly look for a benefit in terms of sterilizing immunity, but if it was missed, we still want to see the potential benefit of avoiding chronic infection. So then we've discussed this question of time. What defines chronic infection? And the real question here is, how long do we need to leave someone infected before rescuing them with the AA therapy to avoid missing spontaneous clearing? And here there's a balance. If we treat too early, we may abandon a good vaccine that was going to lead to clearance, but we treated the people prematurely. If we treat too late, then we expose participants to a longer duration of infection, which has potential consequences. So typically, textbooks define chronic infection as lasting longer than 6 months, but a probably better and more physiologic or biological definition of chronic infection would be the transition to immune exhaustion, after which spontaneous clearance is extremely rare. The problem is that measuring immune exhaustion in real time is very difficult. So we really do need to decide on a standard duration of infection for CHIM or vaccine trials that really balances this idea of missing late clearance against duration of infection. So we looked at this, and a big thanks to Dave Thomas, who really reached out to a lot of different cohorts to try to get some data on this. There's actually fairly little in the literature on this. And this is because the precise timing of infection is rarely known. People often have multiple exposures, and which exposure led to infection isn't often clearly known. And there are few cohorts where they've had standardized and frequent sampling of HCV RNA over time. So Dave actually pulled together data from the VIP trial, the vaccine trial, as well as the BBASH cohort, a prospectively followed cohort of people who inject drugs, and the INC3 cohort that brought together a number of cohorts of people who inject drugs that were prospectively followed for clearance. So when you start out, it looks promising. You've got over 1,000 people in these different cohorts, but once you start excluding people for whom you can't answer the question, either because of prior infection, they received treatment, they were in a vaccine study and vaccinated, they had HIV co-infection, or they didn't have frequent enough measurement. You really need at least 2 HCV RNA measurements, and most importantly, you need to know the long-term outcome of their infection. This initial number drops down to only 45 people with solid data. Of these 45, 29 went on to chronicity and 16 cleared by between a year and 18 months after infection. So let's look at what happened here. And so in green, you see the bars of measurement of HCV-R negativity, and in red that they're HCV-RNA positive. And this is looking at 3 months and at 6 months after infection. And so you can see here the clearance group and here the chronic group. And so what you see is that Most people who had cleared by 3 months, they stayed negative. The problem is, so you can see that the negative predictive value for clearance is high, it's 90%, and it doesn't really get any better at 6 months, it's 92%, but the positive predictive value for chronicity, so if you're chronic at 3 months, you only have an 80% positive predictive value of going on to chronic infection. And if you look at this more specifically, that actually 44% of the people in this cohort, albeit small numbers, that were still positive at 3 months went on to clear. So the concern is that although clearly there are significant caveats to these data, they're limited numbers, but the sense is that starting treatment at 3 months will likely miss at least some spontaneous clearance episodes. The consequence of that, if a vaccine moves clearance forward, so it happens earlier, is that you'll underestimate the clearance rate and then therefore overestimate the vaccine efficacy because you may have more people clearing by 3 months. So although this is certainly not perfect data, I think this gives us some pause about this, about being confident at 3 months. Now, the flip side is that as we've commented, the CHIM may also provide really useful data by using a standardized inoculum, not having to worry about reinfection, confounding the outcomes. This actually may allow to get better predictive models over time, so that with time, we might actually be able to more reliably shorten the duration of infection, at which point we would introduce treatment. But at least on balance, these data would suggest that 6 months would be the optimal time. So let's move to the population. It's critically important to study people at risk for hepatitis C, but it's really important to think about safety first in this model. Safety to the participant and safety to the non-study population, really thinking about the transmission risk. Because of this, at least what we have proposed for initial CHIM studies is we would really focus on a very healthy population, young, with really no medical problems, no underlying liver disease, no substance use because of the risk of transmission, important notification of intimate partners and use of barrier contraception to prevent transmission within the household, and agreement to take treatment. For subsequent CHIM studies, it really is important to document both safety and effectiveness of any vaccine in the population at risk, particularly people who inject drugs. This question of reinfection is always going to be a concern, and of transmission of the CHIM inoculum. So one possibility might be to consider including people on stable opiate agonist therapy, or also just considering looking at real world effectiveness, but it will be critical to think carefully about these decisions later. We talked earlier today about participant compensation and consent, and I think it's important to distinguish this from other challenge studies because of the duration of infection we're proposing. As Jake nicely illustrated before, the Shigella inoculum was confined to hospital for 2 weeks, which I'm sure was very uncomfortable, but it's still only 2 weeks. So we've had a number of discussions about this, and there's clearly no right answers. You get different opinions from different people. We've talked to ethicists. We have generated a community advisory board to give us some insight that includes people at risk or with lived and or living experience of Hep C, and also people with experience in challenge studies. What's clear is that we need a test of comprehension for all participants. We discussed that a bit earlier. It's clear we need to compensate people for the time of study visits, so an hourly wage and also compensates them for travel and other costs. And then what has been proposed is this concept of a weekly stipend for the duration of infection through to the end of treatment to acknowledge the quality of life effects of Hep C infection. And we would propose to use this type of model. Now, as Ray alluded to, the design would really be a multi-stage approach where the initial establishment of the inoculum, so we've talked about really generating an acute inoculum to be used for future CHIM, and how many genotypes is debatable, but probably at minimum of genotypes 1 and 3, and then maybe others. We would have these initial sentinel cohorts that really confirm reliable infection, that you get an acute non-severe hepatitis, and you get reliable and truly universal cure with the AAs. Once we've done that, then we move on to, we really need to understand the spontaneous clearance rate for each inoculum, because that's going to be the benchmark against which the vaccines are compared. And only then do we actually propose to vaccinate people prior to infection and match them to the spontaneous clearance population in terms of these factors that are associated with spontaneous clearance. So Ray sort of showed this first establishment of the inoculum model, where you take HCV either from a chronic donor or from this RNA LNP, and you establish this in one initial recipient who then turns out to be your donor for your subsequent CHIM, for the subsequent CHIM. So this person, upon becoming highly viremic, would be plasmapheresed, and then this serves as the acute inoculum for CHIM, at which point this person would go on to receive treatment, and this would be replicated for the different genotypes being studied. The primary endpoints of this initial establishment would be to really establish the model. So you get reliable establishment of viremia. We really want to see everyone develop viremia, and have some definitions for that they do develop an acute non-fulminant hepatitis, so there are no cases of fulminant acute HCV, and that we get universal cure of infection, certainly with 2 courses, if not on the first course of DAAs. So we discussed a little bit about the acute versus chronic donor. I think Ray really went through that very carefully. And really it comes down to this issue about the inoculum size being considered. And there will be, and it will come up, I think, in the panel about other pros and cons of a chronic versus acute donor in terms of the variability or quasi-species population between the 2 scenarios. And I think that's something certainly to discuss. But one thing that's really important here is by having a standardized inoculum, it really does give us a benchmark for comparing all future CHIM studies and vaccine candidates against a really well understood virus. So, and a single donor can provide the sample for all future CHIM studies. So once we've done that, then you go on to these sentinel cohorts. And really, as was outlined early on in the ethics discussion, we really want this first cohort to be a bit shorter, where these people get the inoculum of what we think will be infectious, 3,000 genomes. They're followed, and then they go on to treatment. This is really just making sure that everything goes smoothly. You then go to a longer sentinel cohort, again, small numbers of participants here. If not everybody became infected, we increase the dose here to the inoculum size to ensure that everyone becomes infected. And again, we're following people until, in this case, out to 24 weeks, but we would treat them sooner, and I'll show you what I mean by this, if they develop severe hepatitis or if they or the PI feels it's required. Then finally, we move on to the spontaneous clearance cohorts, and this is really sort of the end of CHIM as just CHIM before we introduce the vaccines. Here you need a larger number of participants, and we would estimate about 24 per genotype, maybe up to 32, and I'll show you the sample size calculations. It will depend on the spontaneous clearance rate, and this population is really trying to establish what is the spontaneous clearance rate for the CHIM inoculum at 6 months, which will be the comparison for the vaccines. And then you finally move on to the stage 4, where you vaccinate people with a candidate vaccine. They will be a similar cohort size to the population in the spontaneous clearance cohort. Of course, there will have been phase 1 studies to prove the safety and immunogenicity of the vaccine prior to doing this. And then people will get the CHIM inoculum, and we will look to see whether they spontaneously clear infection more frequently than the spontaneous clearance cohort. And this will be matched on these key determinants of spontaneous clearance to the spontaneous clearance cohort. So additional endpoints for the studies, there will be many additional endpoints: virological, immunological, clinical endpoints, and importantly, ethical and qualitative studies as well in this unique model. And then, as was discussed early on, that even if these early vaccine candidates are unsuccessful, CHIM has the potential to really inform future vaccine design by really watching this in real time with detailed immunological and virological studies could inform future vaccine candidates. Now, when do we start treatment? And this is something that we have thought a lot about. And again, no right answers here. I think early on with this first stage, we want to be pretty careful. So really significant ALT elevation, or really almost any bilirubin elevation. And importantly, of course, participant request. And in this case, if the person becomes antibody positive, we would treat them. In the later stages, we would propose to actually push the bilirubin elevation to a higher level, to 100 or 6 in American units, and I'll show you why. Because it's important to think about this carefully. On the one hand, we don't want to have someone go on to develop fulminant hepatitis, and jaundice, of course, is a marker of more severe hepatitis, but it's also associated with spontaneous clearance. So if we treat people at the first bump in the bilirubin, we are going to miss spontaneous clearers, and that will really underestimate the spontaneous clearance rate. So this is really a challenge of getting the balance right. We've looked carefully at the data to try and inform this. We used data from the INC3 cohort, which was a collection of multiple cohorts of people who inject drugs with acute or recent infection. And you can see that ALT elevations were pretty significant here. You can see that the IQR getting up over 1,000 in both groups. And then in a prospective study of acute hep C in over 100 patients, you can see that about half of them had bilirubin elevations that would be clinically relevant, getting up to almost 2. And the mean bilirubin in the population was pretty high, around 80 micromoles per unit, or just under 5 milligrams per deciliter. Here, jaundice was clearly associated with clearance, with a marked difference in the likelihood of clearance, and also having a very high ALT above 800 was also borderline associated with clearance. Multiple other studies have looked at this. We really did pore over the literature, and there's no clear threshold identified to predict evolution to fulminant hepatitis or acute liver failure. And there are many people in the literature with these more severe acute hepatitis episodes with ALTs above 1,000, bilirubin above 100 micromoles per liter, with no cases of fulminant hepatitis or liver failure, which gives us confidence in how we came up with those numbers. Now, we have to be thoughtful about this CHIM process, and certainly if it's not working, we should stop. And we have thought about some futility criteria. If we can't establish viremia reliably, this is a problem. We don't want to expose participants if it's not working. If we have any cases of people developing acute liver failure, this has got to be a reason to stop. Treatment failure is a concern, but I think treatment failure is particularly a concern if someone doesn't respond to a second DAA course. If lots of people are failing with the first DAA course, that's a big concern, but if anyone fails a second DAA course, we need to be very thoughtful and stop. And then, of course, if we see transmission of the CHIM inoculum to non-study participants, then that's really a problem and we need to stop the study. We've thought a lot about risk mitigation strategies here with donor characteristics, so no co-infections, no recent injection drug use to avoid unrecognized pathogens, that they would be otherwise eligible as a blood donor, except for being hep C positive, and they don't have cirrhosis to exclude potentially a more virulent strain. We would do the standard pathogen panel and repeat after SVR to exclude anything that might have been in the window period. The virus would be sequenced to exclude significant resistance-associated substitutions, and importantly, the donor of this virus will be treated with the same regimen to be used in the CHIM studies and only will be used to infect a participant after confirmation of SVR. The virus will be stored under GMP conditions, and we'll have serial testing to confirm stability and infectivity. And then there will be this stepwise evaluation with small sentinel cohorts with sequential enrollment. And after this goes well, then we do need to pick up the pace and do parallel enrollment. There will be a data safety monitoring board with careful oversight. And then, as discussed previously, participants will be offered long-term follow-up. In terms of sample size, it's difficult to be certain here. The sentinel cohorts, there's no specific sample size. These are small cohorts that just really give you a prediction of reliable infection and allow you to think about dose escalation. But the spontaneous clearance cohorts are important because we want to develop a relatively robust estimate of the clearance rate. And we do need to consider these factors that are associated with spontaneous clearance. The vaccine cohorts will really depend on the goal. If we're trying to de-risk a candidate to justify larger trials, that's going to be a pretty small trial to show us an efficacy signal. If we're looking at trying to actually get approval based on a CHIM, then of course that's going to be a larger trial. But the spontaneous clearance rate is going to greatly influence the numbers required. So if you look at this, this is a bit of a complex figure, but just to show you, this is the sample size we would require. This is the estimated clearance rate without vaccination. With vaccination, and each of these lines with the different color squares represents the clearance rate without vaccination, so the spontaneous clearance rate. And this is estimated to give you us 80% power. So if you look at a spontaneous clearance rate of 20 to 30%, which is usually what's quoted, then you need about 10 to 20 participants per inoculum to show vaccine efficacy of 60 to 70%. And I think anything less than that, probably people wouldn't feel is worth pursuing. But it becomes more difficult if you have a spontaneous clearance rate that starts to go up. If you end up above 50%, then you start to need larger cohorts. And remember, these need to be replicated potentially for each genotype to be able to show efficacy. And that's a concern. So this is something that will evolve over time, but hopefully we'll be able to use relatively small numbers to generate a robust estimate of the efficacy. So to summarize, development of a successful CHIM requires a stepwise approach to establish the inoculum and determine the spontaneous clearance rates. Clearance at 6 months balances the chance of missing late clearance with the potential risk and practicalities of longer infection. Initial CHIM studies will be performed in young, healthy populations, but it's critical that we have subsequent evaluation in at-risk populations, especially people who inject drugs. And we need to really be thoughtful about risk mitigation strategies at all stages. This is only one HCV approach. The final protocol will certainly require regulatory input and guidance, but this is sort of a consensus from the workshops of at least one strategy that we could take, and certainly looking forward to discussion and questions about this approach. Thanks so much.
All right, well, thank you, Jordan. Thanks for taking us on the journey to figure out what we need to do for a clinical trial. So let's move on to the last talk, which is probably a little more straightforward, related to how do we leverage this particular model for basic science advances. Naglaa, who is a professor of medicine at University of Montreal and University of Montreal Hospital Research Center, she will share with us her work together with a few other people about what type of basic science studies we can include in this CHIM model. Naglaa?
Thank you, Jake. I'm trying to share my screen. Okay. Is this working now?
Yes, it's fine. You just have to slideshow.
Okay. Do you see the slideshow or?
Yes.
Okay, perfect. Okay, well, thank you. So I'll just try to present a brief summary of the article that we published as part of the supplement with a lot of input from the community, but primarily work with Andrea Cox and Chris Walker. So thank them for their hard work on this. So just to remind everybody, HCV, when the virus enters the body, it initiates innate immune responses like interferon gamma, NK cells, other activities. But one of the determining or one of the key determining immune mediators are CD4 T cells that actually help CD8 T cells that produce antiviral cytokines and have a cytotoxic capacity to kill virally infected cells. But also CD4 T cells help B cells that produce antibodies that are able to neutralize the virus and limit the infection. And they mediate this through different types of cytokines. Also, CD8 T cells, what we know from the work of many distinguished immunologists in the field is that you need a strong polyfunctional, meaning cells that can produce multiple cytokines and have cytotoxic activity in order to control the infection. And the immune response occurs in the liver, but we can also detect it in blood. So blood mirrors what happens, some of what happens in the liver, but not all of what happens in the liver. And that's very important when we consider vaccination. So to go to this CHIM trial, so we assume that there will be 2 trials, or actually the first trial, which is more of a control group, which is to establish, not the one to establish the inoculum, but basically stage 2 and 3 that Jordan mentioned, that are the sentinel cohort as well as the spontaneous clearance cohort, and that is experimental infection with no vaccination. And then the vaccination group, which is stage 4. These 2 groups represent a unique opportunity to understand really the early events during acute HCV infection, because when we work with humans, unlike chimpanzees, we never really know when they were infected unless you are working with people who got infected through a needle stick, but also to understand what happens post-vaccination and whether this response is protective or not. So what we would predict is that in a context of the first group, which is CHIM without vaccination, this will resemble what we would see with primary infection, where there will be high levels of viral replication. The immune response will be delayed, induced maybe around 6 to 8 weeks. Similarly, antibodies would arise around that time. So this is a relatively long time frame. In contrast, somebody who is vaccinated, we expect that this person will already have some memory T cells and antibodies that actually will act very quickly, will blunt viral vaccination. And this is similar to a situation that we see in reinfection studies. So obviously, the kinetics are not going to be the same in both cases. And this is something that we really need to consider as we look at sampling and when we should examine the immune response. So definitely CHIM with vaccination, we expect to see immune responses early on. So we need to have sampling really quite frequent during that period versus CHIM without vaccination. We would expect that this will take a lot longer, and we need to focus our sampling more around that time. And we need to look into blood, if possible, fine needle aspirates to look in the liver, and try to get large volumes of blood through leukapheresis. So these are issues to take in consideration. And I'll come back to the importance of having standardized operating procedures for sample processing and biobanking, because these are unique trials and we really need to do this well. There are many, many things that we can look at. We can look in the plasma for metabolomics, multi-analyte profiling. We definitely can look at antibody responses, neutralization, virus sequencing. Again, fine needle aspirates from the liver, lymph nodes can be used to characterize the immune response, or more complex omics assay like single-cell RNA-seq, ATAC-seq, or others. But of course, we cannot do all of this at once. So we decided that we need to divide this into what we would call first-line assays that are essential to assess the immunogenicity and the efficacy of the vaccine versus second-line assays that are optional, that from which we will learn more about what happens with the immune response. So in the context of T cells, we really need to look at the frequency by looking at the cells that respond to hepatitis C antigen producing interferon gamma, IL-2, IL-21 through ELISpots, fluorospots. We need to do proliferation assays. We need to understand the polyfunctionality of the cells, what cytokines they produce, and whenever possible, to use MHC class 1 and 2 multimers that allow us to visualize these cells and characterize them better. Second-line assays would be more in-depth, high-resolution phenotypic and functional analysis. A lot of the omics assays, like I mentioned, PCR typing in order to track the expansion and contraction of particular clones. For antibodies or B cells, we definitely need to look at ELISA. We need to do ELISAs to measure antibody titers. Neutralization, for that, we need to use a standardized reference set of HCV pseudoparticles or infectious virions. And now with the developments by several groups, we can visualize B cells using E2 multimers. So we think that these are important first-line assays, while second-line assays would look more deeper into the antibody response by looking at E2 CD81 binding inhibition, repertoire analysis, isolation and characterization of the different antibodies, as well as omics on HCV-specific B cells. What is important to note is that one size does not fit all. So we need to be flexible and adaptable. We need to adapt to new findings in the field and other fields in viral, other studies in viral immunology. We need to adapt to the nature of the vaccine being tested. So if the vaccine is primarily generating antibodies, it's different from one that is targeting CD8 T cells or CD4 T cells, although we think that most vaccines will be looking at both, but the kinetics may be different. So this can be informed by the phase 1 trials of these vaccines so that we can adjust our sampling interval and what we need to look at. Finally, we need to have standard operating procedures, especially for sample collection, cryopreservation, because that will be like a huge resource for everybody. We also need to have SOPs for various protocols. It is also important to have reference samples or reference labs that would be responsible for either standardizing these protocols or providing samples that they know what the result is that can be included in the different assays. It is also important, and this is for the whole group, to think about, as this goes forward, to establish a steering committee to prioritize the study, the analysis that will be used, and also to manage sample long-term storage and access, who access what and when. So thank you, and I look forward to the panel discussion.
All right, great. Thanks a lot, Naglaa. We are running a little behind, but we should just go ahead with the panel discussion, which I'm sure will be very lively and dynamic. So Naglaa would be the moderator of this particular session, or the last session. So please proceed, Naglaa.
Okay, well, I'm lucky to have actually all three Nobel laureate on this panel, Dr. Harvey Alter, Dr. Charlie Rice, and Sir Michael Houghton. And also we're happy to have Dr. Ellie Barnes from Oxford, Dr. Julie Bruno from University of Montreal, Jens Buch from University of Copenhagen, and Ray Chung from MGH. And also from NIH, importantly, we have Dr. Emily Erbelding, who we hope to tackle about funding for such studies, Dr. Marion Major from FDA, and Jordan, Dave, Jake have already been introduced extensively. We also have Dr. Mark Ghani and Dr. Mark Sokowski, prominent clinicians as part of this panel. So thank you all for joining this panel. And maybe I will start right away by actually addressing my first question to Dr. Mary. Oh, sorry.
Naglaa, you do both, Paul? Yeah.
Yes. I forgot to mention that we have the poll questions. Thank you, Jake, for reminding me. So you can see them on the screen now. So you need to answer yes, no, I don't know. The first question is: sterilizing immunity, prevention of infection for HCV, a realistic goal for an HCV vaccine? The second question: if sterilizing immunity, prevention of infection cannot be achieved, should a vaccine that prevents chronic HCV infection be the goal? And the third question, if prevention of chronic HCV infection is the goal, how long should we wait to treat infected CHIM volunteers with DAA? So please vote. The poll will be open during the duration of the panel, and Jake is going to announce the results afterwards.
You may have all noticed that the hosts and panelists can't vote.
Yeah, so we cannot bias the opinion. So, yeah. Okay, so we move on to the panel. So we start with Dr. Marion Major. And I think the question on everybody's mind is what would be required by a regulatory body like the FDA to approve the CHIM trial concept for testing HCV vaccines? So I think the first part of the question is CHIM by itself. What is required? So.
Yeah, thanks for that really easy question, Naglaa. So I think actually this whole presentation today, I think, has been excellent. And what I think people have is a very good hold of what the FDA would want to see. We want to be assured of. Let me say first of all, we have had no discussions in the FDA on this. I'm not talking from an FDA guidance or thought. This is my personal perspective. But honestly, we have not discussed this within the FDA. Everything is open for discussion and sort of argument, persuasion. So let me say that first. But I think the first thing is the safety aspect, which has been talked about a lot. It's just basically, can you infect people and cure them without any long-term safety issues? So that's the primary question. And I think that people have a very good argument for that. And I think you have the best experts to be judging that. And then the second is, which type of people would you do this in? I think initially it would have to be healthy people. And I think it'd be a small group of people. I think Jake touched on early on the sorts of things we'd want to see in terms of like toxicology, preclinical data, showing that whatever the challenge virus is susceptible to DAAs. And Ray gave a great presentation on the different options. And I think all of the questions that the FDA would have, I think are being touched on right now. And to say which one of them or which of the answers the FDA would want to have, I can't say. One thing I would say though is, I think that there are going to be a lot of false starts with this at various stages. And so a lot of discussion has gone into this in terms of developing the inocular, but that may end up being stalled because you might test something, you might come across something that you think is right, but it's not because it doesn't respond to DAAs in an animal model or something. So I feel as if the discussion is great, but there has to be some sort of experimental thing has to start at some point. You've got to start looking at things and making decisions and testing them in vitro or in mice, so that it helps make future decisions. There's no magic pathway that can be decided ahead of time at this point, I think. So I really think that there's a lot of really good discussion, and I think most of what the FDA would want is already being addressed.
Okay, well, that's great. So maybe Jordan Feld, you can update us. We've had some discussions with Health Canada, so if you can briefly summarize this.
Sure, I'll just very briefly. We've had 2 meetings with Health Canada, sort of with what we call a pre-CTA, which is equivalent to a pre-IND meeting, to discuss protocols, really following pretty much what I outlined in the presentation. And the first one was received with a lot of questions, and I think the second time we met with them, we really addressed a lot of their concerns, and really I think all the things that Marion pointed out about really looking at safety and really being thoughtful about the process. But they really, I think, were receptive to the concept. I think they understood the unmet medical need, and I think they want to see, as Marion outlined, that we do this in a safe and thoughtful way. But if we do, that this could progress forward, and we're proposing to hopefully think about trying to move this forward in the not too distant future.
Okay, thank you. So just to go back to the question of the population, so I think from the discussion, it would mainly start with healthy donors. PWIDs have a vested interest in that. So I don't know, perhaps Julie Bruno, you can comment on whether PWIDs could be included and when they should be included in such a trial.
Thank you, Naglaa. And yes, sure. I think a lot of the premises from this question has been answered today in this meeting. Jake said why it's important, and it's important to explain to the volunteers why they should participate in some kind of things like that. We also discussed the thing, and Marianne just alluded to this, that from the start we have to have healthy volunteers without any type of interaction of other comorbidities or other things that unmeasured confounders, but also risk of transmission, which is very important. So what I would say is that hasn't been maybe stated is that we know that we have to involve community, and we have to involve community from the start, not from the start of when we're thinking of infecting them, but from the start of this process right now, so that they see the evolution and what we're doing and why, and they begin to think of why it's important and why it's important for them not only to be participant in a trial like this, but eventually as a community to be ready to be vaccinated with candidate vaccine that would work, because we've seen with COVID that this community is not necessarily the one that is the most trustful in vaccines in general. So I think there is a lot of places where people who inject drugs should be involved, but they should be involved from the start. And they should see that they're not the guinea pigs. The guinea pigs are healthy volunteers and a lot of other people. And eventually, maybe one day sooner will be integrate a part of these people that will be interested in being involved in this organization. So I think it's a step-by-step and multi-pronged approach.
Okay, thank you, Julie. So I think this is a good segue to the next question, which I will address again to Marion, and I'd like to have input from Mike and Harvey, as well as Charlie. Assuming that CHIM gets approved as a concept, what would be the essential preclinical benchmarks that the FDA would require to test a particular vaccine in a CHIM trial? Let's say we have like 3-4 candidate vaccines. What is it that you're looking for? So Marion, briefly.
I think it wouldn't be necessary. It wouldn't be the FDA that decides which one gets tested. It would be whoever's sponsoring it. We'd want to see good phase 1 data to show that there's a good immune response, and maybe there may be more of a requirement to look at several of the immunological markers that you laid out, Naglaa, rather than just looking for simple immune responses at a very low level, like is it antibody positive, is it neutralizing antibody, is it T cells? Maybe we'd want to see something that's a little bit more detailed before testing that in a CHIM. That's my thought, but it would be a phase 1 trial.
Okay, thank you. So Mike, you already have a vaccine that passed a phase 1 trial and was tested in chimps. What do you have something to add to that?
Yeah, well, as Marilyn said, regulators would want to see the phase 1 data. And in terms of the immunogenicity endpoints of that, personally, I would recommend that the recipients are eliciting broadly cross-neutralizing antibodies against most of the major genotypes of Hep C seen around the world. I feel that those antibodies should be targeting several linear and discontinuous epitopes that are conserved among the HCV genus. As an extreme example, you wouldn't want to see neutralizing antibodies and they're all targeted to the N-terminal hypervariable region of E2, right? Because obviously we know the virus can evade those very, very quickly. So, Graham raised the issue earlier, how cross protective do we expect a vaccine to be? I guess I would say we would want to see at least partial in vitro neutralization of most of the strains of Hep C seen around the world. And in the trial that NIH sponsored many years ago, when I was in the US, where we just used E1, E2 recombinant antigen with a pretty weak adjuvant called MF59. We did publish in 2013. John Law in my lab assayed for cross-neutralizing antibodies, and we got evidence, surprising evidence, to be honest, that we could neutralize at least partially most of the clades seen around the world. But I think you don't want to just look for cross-neutralization. I think you've got to understand the epitopes that those antibodies are targeting. And there are some good linear cross-neutralizing epitopes, and there are plenty of cross-neutralizing epitopes that are discontinuous. And, some of those require both E1 and E2 to interact to create those epitopes. So, that's why personally I've always favored the heterodimer of E1 and E2 as the preferred antigen. And then, comments around the cellular immune responses I would like to see in a phase 1. I'd like to see, obviously, T helper responses, as you pointed out, Naglaa. But I would like to see multiple T cells responding to the vaccine that are restricted by multiple MHC class 2 alleles, and that shouldn't be that difficult. And talking about CD8 positive cytolytic cells, I think, I think both for CD4s and CD8s, in my opinion, it's important not just to see those types of T cell responses, but to actually see them reacting to epitopes that are highly conserved amongst the genus, because, if you use a big target antigen, a large non-structural polyprotein, for example, there's a very good chance the immunogenicity will be dominated by T cells to variant epitopes. And we know the virus can evade those. So I personally, planning next year, I would hope to see CD4s and CD8s to highly conserved epitopes amongst the genus. And with CD8s, which will be probably the bigger challenge than CD4s, I would want to see those CD8s, the conserved epitopes, being presented by multiple class 1, MHC class 1 alleles, and to be confident that in the population, eventually the vaccine is going to be used in, the majority of the population has the MHC background, class 1 and class 2, to present those epitopes in the majority of the population.
Thank you, Mike. Harvey, do you have anything to add?
Yeah, it's hard to add to all this information that's been out there. But so first I want to say, I mean, this has been so well thought out. Both the publications and this symposium today, workshop, whatever this really is. The presentations have been so well thought out. So we're already very advanced in this CHIM process, which I think is an essential process. I think of all the issues complex issues at every stage, choosing the inoculum is probably the most difficult. And I think it gets down to, do you want to use a patient sample, which might be ideal, acute phase patient sample, but that would have to be identified. Do you want to get that acute phase sample from an actual volunteer, infect somebody so they can be the challenge inoculum themselves? And the alternative is to put out a feeler to ASLD, EASL, all the members, and say, if you identify a case of acute hepatitis C, this is what we want you to do. We want you to get an apheresis unit. We want you to get it in this condition. We want you to store it in this way, because you only need 1, 2, 3 acute cases to have your inoculum. Had Hutchinson still be alive, he would be one of those donors. I think we need a son of Hutchinson.
Okay.
The other alternative is to use the clone virus, where you have all the genotypes very well characterized. And I think Jens Spook needs to talk about that, because this is another viable approach, and the one would have to prove that a clone virus would be equivalent to a human primary infectious inoculum.
Okay, thank you. And Charlie, would you go with a clone rather than an infectious inoculum?
Well, I mean, I think well, we know a lot about 877, so I am kind of a fan of Harvey's gold mine. And I think, you know, the sort of lipid nanoparticle sort of inoculation strategy could, you know, very much allow us to sort of create sort of a good challenge stock in a human volunteer. So I kind of favor, you know, that approach, although I think, you know, we could certainly explore many of them. And I mean, Harvey's right. I mean, I guess when these acute cases come up, that is an opportunity, but I don't know, you know, we would still have to worry about the source. You know, we couldn't pre-select, you know, the person who is going to produce the challenge stock. So, you know, other viruses, other things would have to be sort of ruled out. So I favor, you know, maybe starting with a molecular clone. I think the issue of quasi-species complexity is something that we're not going to really be able to address, you know, sort of in the challenge stock, because if you infect, you know, somebody with, you know, 3,000, you know, sort of genome equivalents or whatever, you're not going to be capturing, you know, sort of the complexity that there might be in, say, a chronically infected, you know, individual. And of course, once that gets into an individual and starts amplifying, there is going to be, you know, sort of quite a quasi-species being created in vivo. So, you know, if you really wanted to try and cover the quasi-species landscape, then you would be going with higher inoculum, you know, rather than something that has just been shown to, you know, sort of infect in the chimp world, you know, sort of 100% of the animals. So I think there are a lot of variables at play here, but I do think that even just sort of choosing one genotype, one inoculum, and, you know, sort of really setting up this CHIM resource, you know, has great value. But I think that Michael's on, you know, the other bottleneck that we face is really sort of getting phase 1 data. Because, you know, there are so many different attractive ways of vaccinating people these days with, you know, whether it's lipid nanoparticles or adjuvanted subunit vaccines or whatever, a lot of things that you would like to try. But, you know, you need that phase 1 data, which I'm sure, you know, sort of Mike and others can comment on this, but it's not, you know, it still requires quite a few resources to sort of generate that database that sort of Marion and the folks at the FDA are going to want to see. And we're going to want to know, you know, as individuals as groups trying to develop vaccines, you know, which ones really fulfill the criteria that Mike just outlined. So anyway, I'll stop rambling on, but I mean, there are still some challenges here, but setting up the CHIM model will be helpful. But it doesn't solve all of the barriers that are there to sort of getting to the finish line.
Okay, thank you, Charlie. So there are a couple of questions on the chat with regards to your comment, Mike, about the discontinuous linear epitope. So perhaps you can answer that on the chat. But just quickly to finish with, because I'm conscious of the time, should we aiming for a broad vaccine to start with? Ellie, perhaps you can address this.
Yeah, Naglaa, you mean a vaccine that will target multiple genotypes?
Yeah.
When you say a broad vaccine.
Yeah.
Yeah, I'm open-minded about this. I think there is a place for more genotype specific vaccines in certain parts of the world. Egypt's a pretty good example. Parts of Asia, you know, where the genotypes are definitely more predictable. I think in, you know, the States and the UK, where we've got a mixture of genotype 1 and 3, which are genetically distinct as you get between the genotypes, we will need vaccines that that target multiple genotypes. And, you know, to be honest, if a vaccine targets genotype 1 and 3, it probably will work against them all, because it will be working against the segments that are conserved between 1 and 3, and therefore between most of the genotypes. So I'm open-minded to this, but I think it would depend on the target population. And you might take different approaches for antigen design for sure, depending on your target population.
Okay. If there's anything anyone else would like to add, you can raise your hand, your virtual hand, before I have one final question. Okay, so Mark?
Yeah, thanks, Naglaa. I just wanted to follow up on, this is a question, again, either for Harvey or Charlie to address on this issue of the viral quasi-species, and what are the relative, benefits and downsides to, perhaps using a sample that's enriched for viral quasi-species? Because I guess if you're going to use a sample from a patient with acute HCV infection, that's more likely to be a homogeneous viral species, whereas from a chronic patient, it's more likely to be enriched with quasi-species. So I'd like to just hear what are the potential, upsides and downsides to each of those approaches, and which would be preferred.
Harvey, do you want to tackle that? You already had your hand up as well.
Charlie might answer this better, but it is a balance. I think it would be ideal to have a fully developed quasi-species in a chronic patient, but that has to be balanced against the advantages of an acute phase patient that is not immunocomplexed and that reaches higher titer and is more infectious. So I think that's a consideration. And I think that has been considered, and in balance, this group has leaned towards getting an acute phase, single, more limited, more monoclonal type of inoculum. The point I was trying to make is maybe I'm missing something here, but we are not developing a vaccine. We are developing a model to test the vaccine. So I think we have to be ready, no matter what the manufacturers might come with. Will our CHIM model be able to say yes or no? This is a vaccine that should go or not go. So it's a little different perspective on what our role is compared to if we were a lab or a manufacturer actually making the vaccine.
Okay, thank you. Ellie, you have something to add, and then Jake.
Yeah, I mean, I've got 2 points. I'll try and be quick. One is that there are multiple groups now trying to make HCV vaccines across the world. And even if we establish successful CHIM models, we're trying hard in the UK, you're trying in Canada, there will be relatively limited access to those models for the number of vaccine candidates potentially coming through. And I do think we need, as a kind of group, to be having very open ways of assessing immunogenicity and comparing vaccine immunogenicity before moving onto the CHIM. And that would mean us kind of agreeing to have standardized panels, probably in an independent lab, to assess broadly neutralizing antibodies in the way that Michael described, and T-cell immunity, so that we can really compare these vaccines head to head before they move through to CHIM studies. And we should be talking about this in parallel with the CHIM, otherwise, because that's going to take some time to set up and a lot of agreements. Justin Bailey and Thomas Peachman have these standardized panels, but people will have to submit their plasma and serum for testing and be prepared to fail in that process. So that was one point. And I would also like to challenge the 3-month, 6-month endpoint that was so beautifully argued for, because there is I actually thought a negative predictive value of 90% and a positive predictive value of 80% at 3 months wasn't bad. And there's significant advantages to a shorter endpoint before you treat patients, including the fact you can get more patients into the CHIM, which allows for more heterogeneity in your population. Lots of other things matter, interferon lambda, gender, and you can test more vaccines, obviously, if you've got a shorter duration of, infection and people are in these kind of studies for long. So I do think there's other arguments to be made about the duration of infection, which could be further debated here, because those actually predictive values were not so bad for 3 months, in my view.
Okay, I see Jordan shaking his head quickly.
Yeah, I mean, it depends a little bit on how you look at it, because it's true. It actually depends on the chronicity rate. So the positive predictive value was really because the chronicity rate was relatively high. So the positive predictive value overall of chronicity was decent, but the bigger point was that the 44% of the people who were positive at 3 months cleared. And that's not quite captured in the positive predictive value. So it's not like, it does depend a bit, and I do think it can give you a bit of a false sense of security, because it's very likely that a vaccine will shift clearance earlier, which could have more people clearing before 3 months and sort of give you a spontaneous clearance rate which is falsely low. That's the problem. So it gives you the impression that the vaccine works better than it does.
Okay,
we can chat about it more. I mean, I agree with you. There's debate here for sure. It's not clear cut.
Yeah.
Just to be clear.
Okay. So maybe final comments from Jake and Marianne, because then I have one last question for Emily. Jake, quickly.
Yeah, I just want to quickly go back to the homogeneous inoculum as in acute phase serum versus quasi-species in chronic. I think, as Ray mentioned, that if you look at those recipient of acute infections, they tend to be a bottleneck. Very few quasi-species actually transmitted or established infection in those acute infected individuals. And point number two is that I think people tend to overemphasize quasi-species. I think quasi-species is a result of chronic infections, right? It's probably had nothing to do with sort of the pathogenesis of acute infections. And then also study from like many of the CHIM studies using monoclonal inoculum, you get the same natural history of in chimpanzee, I meant. So basically the chronicity, pathogenesis is really not that different, right? Between using sort of a homogeneous inoculum versus something have more quasi-species. So I think that's the point that I would certainly be in favor of. There's nothing wrong with using a homogeneous inoculum. It's either in acute phase serum or in molecular clones. So.
Okay, thank you, Jake. Marianne, final comment?
Yeah, I would follow on from what Jake said there, is that I do think that whatever the inoculum is that's chosen, I think a case is going to have to be made that it is representative of a natural isolate. And that can be either through sequence analysis of acute phase. I do think that some of this data needs to be generated and should be shown and used as an argument for whatever the inoculum is. But the other thing is that, I mean, are most infections from chronically infected people? So is there going to be a need to show that what you're going to be challenging with in the CHIM after vaccination is representative of what is going to be the infecting virus in just a general circulating population?
Well, I was going to chime in. Perhaps Julie can follow in terms of following these large populations. So perhaps the Hopkins people have. My understanding is that actually a lot of community-based transmission very often occur in a circle of friends, somebody who had acute hepatitis C and sharing needles, whatever, spread among their groups. So that's actually is another way of how the spread occurs. Somebody who actually had acute hepatitis C end up transmitting a huge number of their friends. So I think that's still, it's not clear whether it's really chronic infected individual has been the sole source of transmissions, right? I think that's an argument.
Yeah. Jake, based parallel to that was if you look at sexual transmission of hepatitis C, it, I think, virtually only occurs with acute infection, or because we have followed many chronic, over 100 chronically infected people and their sexual partners with no transmission. So the chronic phase is less infectious, maybe because of complexing, maybe other factors, lower titers, but I think chronically infected people are less infectious, at least in that sexual model, and probably in the IV drug use model as well.
Okay. And Julie, would you like to add something?
No, I agree. And there are some evidence from network analysis, that is the clusters of individuals that in the acute phase of the infection that drives the transmission. Right?
Okay. Thank you. Kim, do you want to add something?
We definitely have shown that acute infection is more that period that people are more likely to transmit, who are people who inject drugs, compared to chronic infection. We've shown that in our partner studies. But interestingly enough, we've also shown that these folks are they change their partners and sexual networks at a very high rate. So it, it's like a washing machine out there. Sometimes it gets out plenty of times. Half the people who get infected, you can't trace who they got it from, because it's not one of their named partners. We've shown that too. So, but acute infection is definitely more infectious time for transmission.
Okay. Thank you, Kim. Mike, final comments?
Just wanted to mention Fred Prince's old studies. He used a chimpanzee to determine the infectivity titer of like half a dozen chronically infected patients. And he found, and this was published in a review back in the 80s, so he found the infectivity titer to be somewhere between 100 to 1,000 chimp infectious doses per mil. And of course, Harvey's acute phase H plasma is more than 1 million. It's getting on for 10 million infectious doses per mil. So in the chronic phase situation, yes, the quasi-species is much greater, but we also need to remember that most of that virus is complexed with anti-envelope antibody and is neutralized. And, that was shown very nicely by Hichikata that Jordan mentioned in his talk in Bob Purcell's lab. Harvey was a co-author on that, where the infectivity of human samples was inversely correlated to the level of immune complexes. So you can actually measure that, fairly easily in the lab, and you can figure out which samples have the most free RNA, i.e. infectious virus. You can do that for acute, and you can do it for chronics as well. So just a few comments there that hopefully were useful.
Thank you.
Can I just add to that? Because reminded me, Mr. H sample, the highly infectious H77 sample, was on the ascending limb of the ALT curve. It was relatively early in the infection. At the peak of the ALT curve, only like 2 weeks later, that material was put into a chimp and was no longer infectious. And we showed at that point that immune complexes antibody had appeared, immune complexes were present, and it was no longer infectious. So it's only one chimp, but I think that proves the point or supports the point.
Yeah.
Well, thank you very much. So I guess my final...
Naglaa, can I make just sort of one more point? And
Please,
that is,
I
think in the sort of CHIM models that have been sort of proposed, we're talking about maybe one for genotype 1 and another one for genotype 3. And I think again, I mean, what you really want is a vaccine that is going to work in both of those models. And that would, I think, sort of filter out a lot of this concern about sort of just one inoculum being able to sort of represent, sort of all the complexity that might be out there in nature and be potentially transmissible.
Okay, very good point.
If I could just make one comment, Naglaa.
Yes, Jens.
With respect to selection of isolates, I agree with everything that's been said with acute and also with the clone. I think one advantage of picking the clone is that you can pick a sequence that's came from a patient that's well characterized, and also you can then pick something that's been developed into growing in culture, and therefore that particular sequence has already been studied against all the different antivirals that exist and that are being used in treatment. So we know inherently and not a lot about how that particular sequence will respond to antiviral treatment. And we can also pick one that's sort of representative of how it responds to antiviral treatment. We can, of course, also pick some that responds really well to antiviral treatment. You can even consider changing it so that it's very sensitive to antiviral treatment before you put it in as a CHIM. Because all sequences has sequences that give some resistance to antivirals. You can change them, and you can make a virus very sensitive to any antiviral. You can also do the opposite.
Okay,
so you
well,
get more control if you permit yourself to start that first acute infection with a clonal sequence.
Yeah.
Okay, well, a very interesting discussion. Obviously, the viral inoculum seems to be a major issue. Immunity, we have a lot to look at. Definitely, we need to have consensus on SOPs and how to move forward with that. So thank you. I think the last question, which is the elephant in the room, is about money. And I'm addressing this to Emily about potential funding mechanisms for a CHIM trial to start.
Yeah, sure. Thanks, Naglaa. So NIAID is committed to, it addresses a NIAID priority to develop a preventative HCV vaccine. We did support the VIP trial that was conducted by Andrea and Kim, and you're all familiar with that. That was more of a traditional efficacy trial, though, not a CHIM. We have an investigator-initiated clinical trial opportunity that's on our website. This would probably, what you're describing, developing the model even, might be a relatively high-priced proposal and would probably require, it would require approval by the executive committee of our institute. If it's more than $1 million direct cost in direct dollars per year, that level of approval is required. So we also have a leadership group called the Infectious Disease Clinical Research Consortium, which has their own protocol funds, and they have a lot of vaccinologists that are very experienced in clinical trials and vaccines, as well as human challenge models for a variety of things. We've really focused more on respiratory viruses, but we've done also malaria vaccine trials with malaria challenge, as well as Shigella and norovirus. So there's a lot of experience there. I think, they would have to approve use of their allocated funds in order to conduct such a study. And I think the skepticism that would come in, I think you'd need to be prepared for this, is this really less resource intensive, accelerating a timeline towards approval compared to doing a traditional efficacy trial, such as the VIP trial, which NIAID already supported. The human challenge studies that we do in flu are pretty expensive. I could probably give figures to anybody who wanted to just compare, and they're short term compared to what you're talking about with this 6-month endpoint for chronic infection. So something to think about there, whether you really would be accelerating the path towards FDA approval or any approval by any regulatory body with adding this CHIM step. I will also just add, though, that the phase 1 trial that you'd probably need that data before you went into the vaccine cohort for this. We do have resources for that. That probably would be, if you had a candidate that looked good in preclinical studies, that would be probably an easy thing to get access to our VTU sites, our IDCRC leadership group, or we have other mechanisms too that we can do those, that we can support those types of studies even without a investigator-initiated clinical trial grant. So we're committed to supporting this. I think the idea of the CHIM study accelerating progress there's probably still more discussion to have on that point.
Okay, well, thank you very much, Emily. So I think that concludes this panel. Thanks to all the participants. And with this, I hand over to Jake.
Okay, perhaps first order of business to share the poll results with you. And so there we are. For the first question, is sterilizing immunity for HCV a realistic goal for the HCV vaccine? And 31% said yes, 42% no, 27% I don't know. So it's pretty evenly divided, which is fine. And the second would be, if sterilizing immunity cannot be achieved, should a vaccine that prevents chronic HCV infection be the goal? And the vast majority says yes. So I think this is kind of what we thought would be important to consider if we can achieve sterilizing community. And finally, if prevention of a chronic HCV infection is the goal, how long should we wait to treat infected CHIM volunteers with DAAs? Most people say no for 2 months. 6 months seems to be the most realistic answers. And even some people say 12 months would be okay. And some people say just don't know. So I think this is really reflect the sort of state of the field. I think it's great, and that we can get the audience participation in this and to see where they are. And again, just obviously none of us were included in this in this poll. It will be interesting to poll us to see what we think. But anyway, regardless, I just want to go ahead and close this virtual symposium. First, I would like to thank John Ward, Lindsay Hibbert, and the coalition staff for really putting together this organization, which is a lot of the behind-the-scene work to pull this together. So I certainly want to thank them. And I want to thank all the speakers, panelists, as well as the participants who signed on to this meeting for a wonderful date, for a wonderful program. And so the question is really, what's the next step, right? So your thought about it. I think this is something that actually a few of us have really put together, and to really Jordan's group with the Canadian consortium has really spearheading the sort of the first step, or the stage 1, as he mentioned, about identifying inoculum and testing them. And he already actually got some funding opportunity to start this. So I think that's really the first step to be able to establish or identifying potential inoculums so we can study them, right, to put into the clinic. And then, so that's something that is actively going. Obviously, once those inoculums were identified, we will start doing a human challenge study. And that's something that will likely take multiple centers who will be interested in participating in these kind of studies. And for now, we are just really kind of working among few of the groups that would have expressed interest in this particular model. I certainly know that Ellie's group at Oxford has also been pushing ahead with her program as well. So I think this is all hopefully will come to fruition over the next 6 months or so. So certainly we hope to involve and engage all of you as we continue on this dialogue. And I think as we all know that developing a vaccine really the next holy grail, right? Perhaps the final in what we do in terms of HCV. It is true, we may be the victim of our success. We may, be able to really eliminate HCV together with some of the national plan to treat once we have an effective vaccine. But that's okay. I think that's a price we're willing to pay if we've been too successful to move ourselves out of the business of studying hepatitis C. So having that, I'll go ahead and stop, and thank you everybody for your contribution and participation. And I will turn this back to John to close up the symposium.
Thanks, Jake, and thanks everyone for a very rich discussion. I do want to refer people back to that CID supplement. It's an excellent overview of many of the topics that were discussed in greater detail today. I think we really covered rich perspectives of from a patient perspective, clinical perspectives, and immunologic perspectives, and study design and questions around inocula, among others. In addition to the CID supplement, we'll be putting together a synthesis report to try to summarize this discussion together. As I mentioned at the top of the webinar, the video recordings and the slide presentations on our website at globalhep.org. Thank you all for a great webinar, and look forward to working with you in the future to eliminate hepatitis C. Thank you. And please fill out the evaluation poll that's in front of you.
Right, great. Thank you, everyone.